kinetic radiation in matter will take place, the exchange of energy will take place only when it is matches perfectly. So what happens? The same rule applies here. So when the IR radiation interacts with the molecule, what will happen? The specific applied quantized IR light, which is matched with the internal vibrational frequency of the molecule, because every molecule has got its own vibrational frequency. You have to imagine the molecule as if one of the simplest system, if you want to visualize a diatomic molecule, say atom A and B are connected, and this bond is actually a spring. So this is just a visualization that will give you an idea about the molecule. Now what will happen when you are irradiating it with the light, and that is the light coming from mid IR region? What will happen? It will absorb and it will set into vibration. And due to absorption of the IR radiation, the net change in the dipole moment of the molecule occurs and causes the vibration of the molecule, like stretching and bending vibration. And whatever light is absorbed, the remaining is transmitted, and transmitted light is being detected by the detector, and you get an IR spectrum. So normally this is the scenario. So you irradiate with the sample, and the sample absorbs specific frequency out of the whole range, and it is the rest is transmitted, and the plot is you get IR spectrum in the end. And actually, because of the stretching and bending vibrations, what happens? You get a peak. And it is interesting to note that IR spectrum is usually a plot between percent transmittance and a wave number. There are other means also where you plot between absorbance versus wave number. So in that case, if absorption takes place, you will get a peak like this, right? However, when you are plotting it against percent transmittance, you will get an inverted valleys. You will get a valleys, not inverted valleys. You will get a valleys in the peak. And this particular point refers to the 100% transmittance. 100% transmittance means 0% absorbance. So this is the way actually IR spectrum looks like, and this is the way IR spectrum occurs. Now, one of the most common technique I will not be touching, because I am not from an analytical chemistry, so I don't know much about the instrumentation. However, I will be just touching a few important aspects. The most commonly used IR spectrometers are nowadays a Fourier transform IR spectrometer. And these are actually, how is this working? So you see, this is the source of light, and from the source of the light, which is usually a nurse Glober lamp or Glober source, which produces a mid IR, this will produce an IR radiation, and it is passes through a beam splitter that will split into two part. So this is one, and this is two. So it will split into two part, and from there it is allowed to, so beam of, if the intensity of the beam is split into half of the intensity goes to this moving mirror, and half of the goes to the stationary mirror. And after colliding with the mirror, it again comes back at the beam splitter, and the combination occurs, and then it is subjected to pass through the sample. So means splitting a beam and then again recombining is the principle of the Fourier transform IR spectrophotometer. And it is allowed to pass through the sample, and then as usual, sample will absorb specific frequency, and all other frequency will be reached the detector, and the detector will be transformed into an electronic signals, and you will get a spectrum like this. So this is the normal setup. So you got an IR spectrophotometer which is connected with the computer and giving you an IR spectrum. So this is just an introduction part about the FTIR. Now, one of the issue, like when you are handling, many of you are having the IR spectrophotometer at your lab. It is not a very expensive affair. You can get up to 10 to 15 lakh. So one of the issue that usually handled during my postdoc also, we also got some issues with handling the sample, because you have to choose an appropriate sampling or sample handling material for recording the IR spectrum. Sometimes you can record neat, sometimes you can transform into pellet, and sometimes you have to prepare its solution, you have to dry it, and you have to get a thin film. So there are so many methods for preparing the sample. So if you are having an IR spectrophotometer and you are having a specific sample, you have to choose which method you have to use for sample handling. So in fact, it is interesting to note that nowadays there are some probe-based IR spectrophotometer also. However, the sample handling presents a problem, and there is no material that is transparent over the entire window. So you see, uh, normally you can use NaCl also. NaCl can also be used as a material, but it is transparent above 625. So you cannot use NaCl below 625. You can see the range 4,000 to 40,000 to 625 centimeter inverse. Same way KBr can also be used, and KBr is actually used in the form of a disc. So what we've done, with the help of a dye, you have to mix a specially a dried sample or sample along with KBr, dry KBr, unhydrated KBR. They are mixed in a mortar and pestle and then transformed into a disc, and that disc is kept in an IR spectrometer. So this is having a somewhat larger range, and it is transparent above 4,000 centimeter, 400 centimeter inches. AGCL is also used for my sample, but it has not been very much satisfactory. Again, it is about the same range, 435. It is transmitted above 35 centimeter. Polyethylene cells are used below 600 centimeter inches. So if you want to analyze the IR spectrum below 600 centimeter inverse, then you can use polyethylene cells. And the gases can be scanned in a 10 centimeter scale. Air analysis, the pollutants coming out of the industrial effluents, or the gases that are coming out of the industrial chimneys, they can also be analyzed by the IR spectrometry, and where you have to use a closed tube which can be made up of one or other material, and usually the cell length should be 10 centimeter. Liquid samples are scanned neat, so you can just simply neat. And for polymer, resin, and amorphous solid, films are often formed using volatile solvent. So on the sample holder, the sample is dissolved in volatile solvent, say chloroform or something like that, and then the sample is allowed to it will get evaporated, and your sample will be forming thin film over there. So these are actually some of the means of sample handling which you need to um, identify depending upon your requirement. So I am normally doing organic chemistries. Most of my sample they can be done in the form of KBr. They can be done neat. Neat will be also good. So if you take neat, uh, means without mixing anything, you can just put the sample as it is, and there you can use the thereby you can use the you can do the IR analysis. Now let us come to the one more theoretical part. Let us understand what these molecular vibrations are. Now uh, 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 say a diatomic molecule is assumed to be two atoms connected together by a spring. So let's say the spring is an equilibrium position, and if you are stretching the spring, this is the stretch position, and when you are compressing the spring, this is the compressed position. This is the equilibrium position. So normally when a spring is subjected to vibration, there can be two kinds of vibration. One of them is called stretching vibration, which is usually requires more energy, and therefore they appear at higher frequency, because the basic equation E equals to hc nu bar. Nu bar refers to the wave number. Okay, so even though we are using the word frequency, normally IR spectra are discussed in terms of wave number, but the common uses is they are referred as vibrational frequency. So it's better to call vibrational frequency in terms of wave number. One more interesting aspect about this is when uh, say if you have a spring and you are trying to bend it, you will require lower energy and it will appear at lower frequency. So there are in fact 6 different types of the molecular vibration. One of them is stretching, Another one is asymmetric stretching. So symmetric stretching and asymmetric stretching, I will be talking in more detail in the next slide, and just to give you an idea. And there are again two type of bending vibration. The bending can be in plane and out of plane. Seizing and rocking are the kind of in-plane bending, whereas twisting and dragging are the kind of an out-of-plane bending. And remember that number of bending vibration is usually more than number of the stretching vibrations. And use what is the point to be kept in the mind here? Actually, all the stretching vibrations, they will be occurring at higher frequency, and bending vibrations, they are occurring at the lower frequency. So this is one point that is to be kept in the mind. Now, this is a pictorial representation that will give you an idea about the different types of the stretching and bending. In stretching and stretching vibration, what will happen when you have a symmetric stretching? Both the atoms are away. For example, this is an angular molecule like this, and let us say this is atom A and this is B. A and B are going away by the same distance from the central atom C, or they are coming to C by the same coming towards C by the same distance. So if this happens, what will happen? If this happens, the bond length and bond length, this bond length and this bond length, these two bond lengths will increase or decrease symmetrically. However, in asymmetric stretching, one bond goes away, say A goes away, and whereas B is coming closer towards the central atom C. So these are the examples of symmetric and asymmetric stretching. This is just a representation. So far as bending is concerned, you got scissoring and rocking. Scissoring and rocking, these are in-plane bending. So what happens here? Only in all the bending vibrations, what happens? In stretching vibration, the bond length changes or interatomic distance changes, whereas in bending vibrations, angle changes. Okay, change of angle. So here what happens? The scissoring involves the movement of the two atoms towards away from each other, and so bond angle decreases and increases with time. So what happens in scissoring? It is just like a scissor. You know, everyone is using scissor. So what will happen? So they are coming closer towards you, closer towards each other. So this towards each other and going away from each other. Whereas rocking involves they are both going in the same direction. So this is in plane. They are not, the atom A and B has not changed their plane. They are in the same plane where they are actually. Whereas in the backing and the twisting, which is out of plane, what has happened? There is a change in the angle between the plane of the plane of a group of atom. So what happens in wagging? One of the atom is going below the plane, and one of the atom is coming above the plane. So it is coming out of the screen, and green is going behind the screen. So you can imagine what sort of things are going on. Whereas in twisting, in wagging, so here both will go plus wagging, and in twisting, one of them will go come up. Dr. Padmini ma'am, please mute yourself. Yeah, thank you, ma'am. So these are the different types of stretching and bending vibrations. Important thing as a chemist or as a biological scientist, we need to know that stretching requires larger energy, bending requires lower energy. This is the relation that we are going to use for the interpretation of the compound. Now, if a molecule is undergoing vibration, whether these all vibrations will be absorbed, the simple answer to this is no, because only those vibrations they will show the IR spectrum only when the applied infrared frequency is equal to the natural frequency of the vibration, and only molecule gives the IR absorption. So atoms, you will not get atomic IR spectroscopy. Atoms do not show IR spectroscopy. And what is the condition? Molecule must have dipole moment, and the mode of the vibration of the molecule should not be the center of symmetry, and the molecule should be polar. So what happens? So essential condition is what? So when there occurs a molecular vibration, there should be a change in the dipole moment of the molecule. There should be a change in the dipole moment of the molecule. Say this is a state A and it is being transformed into state A prime of molecular vibration, then the dipole moment mu of this A should not be equal to mu prime. So if this is the situation, it will not show absorption. So mu should not be equal to mu prime, then only it will show IR spectrum. So it is important to note that it is not necessary that for a compound to have a permanent dipole moment to be IR active. The same way, in fact, what is important? There should be a change in the dipole moment of the molecule when the molecule undergoes vibrations. Okay. Now, there is one simple calculation. Normally, a molecule can have, any molecule can have 3n degrees of freedom. Degrees of freedom refers to the coordinates which are required to define the locate the molecule. Say, for example, if you want to locate me, oh, PPT not visible.
Visible, sir, visible. Needs to pin it. It's visible.
Oh, okay. So normally, if you want to locate me, so as Rasad sir said that I am from Khamgaon. Khamgaon means it has got some altitude and longitude. If I want to know my position, it has to be characterized by the three coordinates. So in space, I am characterized by the three coordinate x, y, z. So x, y, z are actually the degrees of the freedom. Now, if I am a single atom, then you can explain my location by the three coordinate x, y, z. But if you have n number of atom, then you need to require how many coordinates to locate the position of all the atom? 3 into n. So these are the degrees of freedom which are required to locate normally. And if these motions they undergo, like translational motion, rotational motions, and vibrational motion. So these 3n vibrational degrees of freedom are actually a sum of translational, rotational, and vibrational degrees of freedom. And our aim is to find out the vibrational degrees of freedom, because this number of vibrational degrees of freedom is somehow related to the peaks that you are going to get in the IR spectrum. Okay, so we want to know how many vibrational degrees of freedom are there. So, for a linear molecule, if you have a linear molecule, for example, this pen. So this pen, if I have a linear molecule like this, atomic molecule like this. Now, if I rotate it around this axis, this will be one rotational degree of freedom. If I rotate it around this axis, this will be second degree of freedom. But if I rotate, there is no change in the position of the atoms bonded here. So let us say A and B. The position of the atom A and B do not.
Excuse me, sir. Audio is not coming. Audio is not there, sir.
Hello. Yes.
Yes, sir. Yes.
There was an issue of the connectivity. I apologize for that. Just a moment. Okay. Okay.
Yes, sir.
Okay. Thank you. So Normally you see for a linear molecule you got 3n minus 5 degrees of freedom, and for non-linear molecule you got 3n minus 6 vibrational degrees of freedom. So let us take one example. For example, if you take a CO2 molecule, in CO2 molecule how many total number of atoms are there? One carbon and two oxygen means 3 atoms. So 3 into 3, it's 9 minus 5, because CO2 is a linear molecule like this. So it is very, that is why it is really important to know the structure of the molecule as well. If you know that CO2 is linear or not, then you can find out the degrees of freedom very easily. On the contrary, H2O is an angular molecule. So here the 3 into 3 minus 6, so 3 vibrational degrees. It means that you can expect 4 peaks in the IR spectrum of carbon dioxide. Actually, you get only 3 there, and you can expect a 3 peaks in the IR spectrum of the water molecule. And one more important point is, not all the vibrational degrees of freedom results in the peak, because sometimes there occurs a combination of, because even though the vibrational degrees of freedom can be more, say in case of benzene, let me give you one example. C6H6. So in case of benzene, you got 6 plus 6, 12 atom. 12 into 3 is 36, and it's a non-linear molecule, so minus 6. So how many vibrational degrees of freedom? 30. So you can expect a 30 peaks in the IR spectrum, but you don't find 30 peaks in the IR spectrum. You find only few, because many of them having the same frequency or very close frequency, they undergo they show the combination band. Okay, but this is just one of you that you can get this much number of signal in the IR spectrum. So let's understand what is the basic equation, mathematical equation that is really involved for the calculation of the vibrational frequency. Based on the Hooke's law, this equation has been derived by using the quantum mechanical calculation, and the vibrational frequency for a particular vibrational transition, it is given by the equation nu bar is equal to 1 upon 2 pi c under root of k by mu, where k refers to the force constant and mu refers to the reduced mass. Reduced mass is actually, say for example, if you have a diatomic molecule with the masses m1 and m2, then reduced mass is m1 m2 upon m1 m2, m1 plus m2. Now here you see force constant. So you see these are the things which are constant here. See, these are all constant, right? The force constant K is actually, it refers to the strength of the bond. It refers to the strength of the bond. Strength of the bond. And you see single bonds are, if you compare the force constant for the different bond, then you will notice that roughly it is in the range of 5 into 10 to the power 15 for single bond. Say this is for carbon-carbon single bond, this is for carbon-carbon double bond, and this is for carbon-carbon triple bond. You see roughly 5 into 10 to the power 15 dynes per centimeter. This is the force constant for single and triple bond. So from this you can see that vibrational frequency is directly proportional to the force constant K, which we can assume to be the strength of the bond, whereas mu, it is inversely proportional to the mu. Larger atoms masses lowers the frequency. So you see, and this can be explained very easily with the help of so these are the values of the K for carbon-carbon triple bond, carbon double bond, and carbon-carbon single bond. Sorry, these are the values of K, and these are the values of the nu bar for the corresponding frequency. So normally you get a carbon-carbon triple bond around double 00, carbon-carbon single bond around 1650, and carbon-carbon single bond around 1200. So this particular equation is really important to know where you are peak due to a particular stretching or bending vibration will occur. Normally, while analyzing the IR spectrum, we focus our attention on the stretching part, because bending is again important when it comes to the real-time application of the IR, because bending can give you a lot of information, which is also referred as the fingerprint region. We will deal with that little later, but let's focus our attention on this particular equation. So nu bar is directly proportional to the force constant and inversely proportional to the reduced mass. And you see how the how the vibrational frequency. So you see this is an GIF file which is used. So this is a H2 molecule and this is I2 molecule. Depending upon their mass, you can see the frequency of. So when I2 is actually getting stretched and compressed, in that time H2 is actually stretching and compressed for more than 5 times. So it means that the vibration masses, when the masses are less, bonded atom M1 and M2 are small, vibrational frequency is higher, whereas when M1 and M2 are larger, then the vibrational frequency is lowered. So this is again one example. So you know, in while knowing the structure of organic molecules or the biological molecules, you normally have carbon hydrogen, carbon carbon single bond, carbon oxygen single bond, carbon halogen single bond, and carbon chlorine single bond. So you see, carbon in this series, carbon is the atom. The only difference is the other atom, like hydrogen, carbon, oxygen, chlorine, and bromine. So what is happening when you change hydrogen to carbon, carbon to oxygen, carbon, oxygen to chlorine, and chlorine to bromine, you are actually increasing the reduced mass. When you are increasing the reduced mass, what is happening? The vibration frequency goes on decreasing, and you can clearly see that because this is an earlier example where reduced mass was same because both the atoms involved are carbon and carbon. But here in this case, two atoms are different, and depending upon their masses, you will notice that depending on the reduced mass, as the reduced mass increases, the vibration frequency decreases. So these are some of the few fundamental equations that one need to keep in mind in order to understand this will be very helpful to know where our peaks will appear. Because while interpreting the IR spectrum, you know the regions where your vibration frequency will appear in the spectrum. Okay. Now, let us understand what is IR spectrum. So IR spectrum is essentially a plot of a percent transmittance versus the vibrational frequency, that too in terms of wave number. Okay, it's already mentioned in terms of wave number. And what is percent transmittance? Percent transmittance is I upon I0. I upon I0 is intensity of a transmitted light upon intensity of the incident light. So when the percent transmittance is 100%, you will see no peak, and here it becomes 0. Okay, so there are two important regions in the IR spectrum. One of them is called as a functional group region, which is normally from 4000 to 1500 centimeters, and the other region is a fingerprint region. So here you will find the peaks due to the stretching vibration, whereas in the fingerprint region, mostly you will find a peak due to the bending vibration. Of course, you can see some of the peaks due to the bending stretching vibrations as well. However, but most of them are bending vibration. And remember that this is a region which is called as a fingerprint region because no two compounds can have the same IR spectrum in this region. No two compounds, two different compounds cannot have same IR spectrum in the fingerprint region. There may be some sort of overlapping in the functional group region, but they will definitely show some sort of difference between the fingerprint region. And stretching mode of vibration, it contains very few peaks in the functional group region. You will see more number of peaks in the fingerprint region, and mostly it is due to the bending vibrations. And you know, bending can occur when we have seen the types of the bending. Stretching was of symmetric and asymmetric stretching, whereas bending were of 4 different types. Okay. So the pattern of peak is easy to analyze because stretching vary within a narrow range, and the pattern of the peak is very complicated due to the complex vibration, which creates a unique IR band to distinguish the compound. So you know, in fact, IR spectroscopy can be helpful to identify the ortho, meta, and para isomer because they show a unique pattern of peaks in the fingerprint region. I have an orthonitrophenol and paranitrophenol, and if they mix IR spectrum, I can definitely distinguish on the basis of the peaks or the nature of the pattern that I get in the fingerprint region. So this is important. And remember that functional group region, normally you get sharp, broad, strong, and medium peak, whereas we get sharp and weak peaks in the fingerprint region. Now, when you try to analyze the IR spectrum, what are the things that you should look at? The first thing first that you should look at is the wave number, at which wave number you are getting the peak. The second type of analysis that you need to do is the shape of the peak, whether it is broad or whether it is sharp. If it's a broad, normally due to the H bonding or the presence of water molecule. Hydrogen bonding actually broadens the IRP. And the third parameter that you need to look at is the intensity of the peak. And it can be of strong, medium, and weak intensity. Strong peak covers most of the y-axis like this. A medium peak covers half of the y-axis, which is like this, and weak covers less than 1/3 of the. So this is actually one of the guideline to know whether which peak is strong, which peak is weak, and which peak is medium. And the intensity of the peak is actually related to the polarity of the bond, or in terms, or in, you can say dipole moment of the bond. The bond moment will decide the intensity of the peak. So, for example, carbon-carbon double bond, the peaks are weak as compared to carbon-oxygen double bond, because carbon-oxygen double bond is more polar. Okay, so difference in the electronegativity is one parameter on the basis of which you can predict the intensity of the peak. Okay, so these are some of the fundamental things, and this is an example how the broadening of the peak occurs because of the hydrogen bonding. Because this is a neat sample of carboxylic acid. In the carboxylic acid, you know, there occurs a broadening of the peak due to hydrogen bonding. Okay, now let us summarize the factors affecting the vibration frequencies of the functional group. Now, effect of different mode of vibration. So normally, you know, stretching occurs at higher frequency than bending. Stretching occurs at higher frequency than bending. Then reduce mass, higher the reduced mass, lower the vibrational frequency. More vibrational frequency, more force constant. It should be constant. And in addition to these, there are other effects like electronic effect. So when there occurs conjugation, conjugation lowers the vibrational frequency. So it is inversely proportional to the vibrational frequency. For example, if you have a C double bond O and you have a C double bond O in conjugation with a carbon-carbon double bond, this C double bond O will appear at lower vibrational frequency because of the resonance. What will happen? This bond will be showing a single bond character. Hydrogen bonding normally lowers the vibrational frequency and it also leads to the broadening of the peak. Okay, so there's one. Then multiplicity of a bond. So more the number of bonds, more will be the vibrational frequency. This was quite obvious from this. So carbon-carbon single bond, double bond, and triple bond. You see the vibrational frequency changes from bar 12, 1200, 1600 to double zero. Okay. And one more parameter which is again affecting the vibrational frequencies of the functional group is the percentage of S character. More the percentage of S character, more is the bond strength, because S character means you are having the more less diffuse character in the atom, and because of that, bond strength also increases, and that is why vibrational frequency increases when you got the percentage of character. And the same is explained on the basis of this. For example, in this case, normally you see when you talk about CH, so sp3ch, sp2ch, and spch. So you see SPCH occurs at much higher value than sp2ch than in sp3ch. So with these fundamentals of IR spectroscopy, now we will try to use few spectra from the textbook and few examples from the research papers. Okay, so let us see. Because see, there are different. If you Google the range of IR, you will find a different images of this. This is one of the images which I have forgotten to acknowledge the source. but I have got it from the web only. So normally you see the region from 3200 to 344 is OH stretching, NH also occurs in approximately the same range. And the way I was talking about CH, this is an example of an SPCH, right? The carbon is in a state of sp hybridization in alkyne. So alkyne, it comes around 3300 centimeter inverse, much higher than the alkenyl. So this is an example of an sp2ch bond. It occurs just above 3000, and the alkyl, which is actually sp3ch, it occurs less than 3000. So it normally comes around 2950 or 2960 or 2920. And this is actually this is supposed to be a CH stretch region. Then you got a triple bond region. So normally here we have seen all the hydrogen stretching region, which normally comes in this range. And there are a few other regions. This is just an overview because we have not included a very specific peak that you get for the carboxylic acid and aldehyde that we will be talking in more detail as we proceed. And then you got a triple bond region. This is actually supposed to be a triple bond region. You get peak due to carbon-carbon, triple bond, carbon-carbon, nitrogen, triple bond. And as usual, you see, you can predict the intensity. Carbon-nitrogen peaks are supposed to be stronger than the carbon-carbon triple bond because carbon-nitrogen bond is polar. So this is how you need to apply the concept that you have studied so far. Thereafter, you also see the carbon double bond stretching region where you get C double bond C as well as C double bond C double bond C as well as C double bond O as well as C double bond N peaks. And the region from you see 500 to 1400, which is often referred as a fingerprint region. It is usually a region that will give you most specific fingerprint of the molecule. Here you see here the word uses gleaming forest of the peak because you see so many peaks in this region. But this is actually very helpful to reveal the identity and the purity of the compound. Okay, so there are different zones, zone 1, 2, zone 3, zone 4. So one of them is hydrogen bonding, then triple bond, H bonding, then triple bond, then double bond, and then single bond, and thereafter fingerprint region. Okay, so OH bonding stretch peaks, they are usually strong and broad and U-shaped, elongated U-shaped. So this is an example of an OH peak. You see, this is a U-shaped peak. Then terminal alkyne, which comes around 3300. So if you get a peak in an IR spectrum around 3300, that peak is broad, then you can say that it may be due to OH. But if you are getting a sharp peak like this, then it means this is a sharp peak. See, this is a very sharp peak. Then it could be because of the acetylenic hydrogen in the molecule. Similarly, you can find out NH. So primary amine usually gives 2 signal, and secondary amine gives you 1 signal. So this is an example. See this sort of peak, right? 2 peak, and here only you see only 1 peak. Okay, this peak. So then comes the CH stretching. So for aromatic compound, out of plane CH bending comes in the range of 900 to 690, and this is actually really helpful to differentiate between ortho, meta, and para compound. Okay. Then zone 2 comes with a triple bond region where we talk about the strength of the carbon-carbon triple bond, double to double 0 to 2000 and double to 60 to double to double 0. Double to 20. So carbon-nitrogen triple bond, it is usually intense and very sharp peak you normally get for carbon-carbon triple bond, carbon-nitrogen triple bond, and carbon-carbon triple bond. Now comes the third region is for carbon-carbon double bond region. So normally aldehyde comes in the range of 1740 to 1720. You can see this is a collection of sword of because one of the important applications IR is in the carbonyl region. So So to identify the carbonyl, one of the best technique is the IR spectroscopy. You see aldehyde, ketone, carboxylic acid, esters, they can all be identified using the IR spectroscopy. So you see aldehyde really at higher number, then comes the ketone, then comes the carboxylic acid, and the esters they again come at relatively higher than the carboxylic acid. So normal range is around 1700. So you cannot keep remember all this value, but the range is 1700 to 1750. So in these range, you normally find the peaks due to the carbonyl stretching frequency of most of the carbonyl functionalities. And this, as mentioned, so as we mentioned, what are the peaks to note at? To know the wave number, to know the shape of the peak, whether it is broad or sharp. And third thing is to know its intensity, whether it's strong, medium, or weak. So the three important characters of every peak is to be analyzed. Then only you can, of the significant peak is to be analyzed. Then only you can arrive at some conclusion related to the structure. Okay, so you see carbon-carbon double bond. When carbon-carbon double bond peaks are there, the peaks are usually low in intensity as compared to the carbon-oxygen double bond. Carbon-oxygen double bond peaks goes way along the y-axis. Huh? Okay, so these are some of the examples. Then carbon single bond region which comes from 100 to 100, and carbon-oxygen double bond in 150 to 1000. So normally, when you see higher spectrum, you see get a peak. This you get C double bond O around 1700, C single bond O around 1200, and carbon carbon single bond around 1100. So these are normally the situation of the peaks. I'm just trying to show you the peak as well, because this will actually give you an idea how the peak looks like in your sample. Because when you are seeing the spectrum from the textbook, it looks very clear, but when you synthesize a compound and when you look at it's a IR spectrum that you get from that you get, then sometimes you find it very difficult for the analysis. Okay, now let me come to the last part for this particular topic, and it will be about the applications of the IR spectroscopy. I have tried to give you an overview, the region, the fundamental concept, the vibrations, the fundamental equation correlating the vibrational frequency. Now I am trying to tell you the one, some of the applications. Thereafter, I will talk about two or three examples from the research paper, how IR spectroscopy has been explored by the researcher to come at to contribute to the science that I will be talking about. One example from forensic science, I will take one from the biological sample and one from the two from the biological science. These are the examples that I will be covering. As I mentioned, so it is one of the technique for identification of the functional group, and I have explained you with the example of a cholesterol. You can actually monitor the progress of the reaction. Nowadays, probes have been developed. So you just put a probe in the reaction mixture and you connect it with the IR spectrum, and then you can monitor the progress of the reaction and what change is actually happening in the functional group. So while monitoring the progress of the reaction, you know what are your reactions, you know the functional group present in the reaction, and you know what is the rate during the progress of the reaction. So you can actually monitor um, uh, uh, how the group changes during the course of a reaction. And you can always distinguish between the hydrogen bonding, which is intermolecular and intramolecular. And the best example, the textbook example, this is orthonitrophenol and paranitrophenol. And these are the hydrogen bonding which can be studied by taking the IR spectrum by changing the concentration. Because if you increase the concentration in the two, because this is intramolecular, distance between the two will increase. As a result, their vibrational frequency will increase, because vibrational frequency decreases with the hydrogen bond. You can study keto inverterism, because in keto you get the keto peak due to keto, as in enol you get the peak due to the OH, and they all together in the spectrum. You can also study cis and trans isomerism, because cis and trans isomerism can be studied um, uh, cis and trans-isomerism can be studied by uh, cis and trans-isomerism can be studied by knowing, say, I can give you an example. So you got, say, this kind of a trans isomer, and you got this kind of a cis isomer. Then in cis and trans isomer, you can easily differentiate because there will be a change in the moment and there will be change in the intensity also. And one more important thing is on the basis of CA bending vibration. So cis isomer gives a peak at around 700, whereas trans gives a peak around 960 centimeter inverse. And you can compare them, easily compare them. Then one more example is you can detect the determination of impurities. If you have a pure iron sample of a pure compound, if you have some sample whose purity is determined, you can just overlap the two spectrum over each other and you can find out whether you have additional impurities or not. If you see additional impurities, impurity. So that is why iron spectrum is recorded deferred as a fingerprint of the molecule. And you can always determine the presence of water in the sample. Okay, so let me give some examples uh, um, from the analysis part. So, what are the things that you should note? Position of carbon, means wave number, intensity of absorption, and shape of absorption. Whether you get a broad or sharp peak. So, position will give you an idea about the energy required for the vibrational transition. Intensity will give you information about the and normally these are the things that you should look at. Uh, Intensity can be strong, weak, or medium. Say if you have a polar bond, then you get will have a large dipole moment, and for carbon-carbon double bond, you got a small dipole moment. So carbon double bond switching, they are usually at with a very medium to low intensity, whereas for the double bond, they are strong. So carbon nitrogen triple bond, yeah, there's a medium peak. Carbon triple bond in ethylene a weak peak. Whereas when you have a symmetric then you find a peak in the higher spectrum. Okay, so this is one example. In addition to these, so some of the examples that I want to highlight over here is you can always differentiate between ketone and aldehyde because aldehyde is characterized by the presence of a peak at around 2.20, a very weak peak which is usually occurs at 2720 centimeter inverse, distinguishes aldehyde from ketone. Now, acid usually show a very broad peak which is ranging from 2500 to 3000 centimeters. So you get a very broad peak like this. And you see the ring size. So when the decrease in the ring size, so if the ring size is decreasing this way, then the carbonyl stretching frequency increases, and you see the normal carbonyl frequency is around 1700, whereas for the cyclobutanone the frequency is 885. And one more important application which I was talking about is careful examination of the field region. In fact, this is difficult task. Why this is difficult? Because this is so you compare the finger you are getting so weeks, isn't it? So orthoisomer shows one strong peak around 750. Meta shows three strong peaks, a pattern like this, three which is separated by around 90 centimeter inverse. Difference of them is around 90 centimeter inverse. Not 90, it's 60, 110, 110. 770 plus 99 only. So difference of 90 is there. And for a para, you will get a strong peak around 800 centimeter inverse. So this pattern can be helpful to distinguish the ortho, meta, and para isomer from each other. Okay, now let me talk something about some examples which are really, this is all about the basics of IR spectroscopy and how to analyze the IR spectrum, why you get a peak at a particular vibrational frequency, what is their intensity, how is the shape of the peak. These are some of the things that one should be vigilantly observe in the IR spectrum. At the same time, you see, as I was talking about, that people have developed a probe. So you have a sample reaction going on and you put a probe in it and you can monitor the progress of a reaction. So what is important is you know the structure of the reactant, you know the possible structure of your product and the byproduct. So you can actually monitor, and this is actually helpful in telling you about the mechanism. So this can help you to probe the mechanism, to provide an evidence in favor of mechanism. So you can use this kind of technique. Some of the companies are providing this sort of IR spectrophotometer. In fact, this is really expensive affair, and we normally do not do this kind of thing in our labs, especially when we are working in an affiliated college, when you have a limited research facilities. So working with that is really, really difficult. But however, people are doing it and you should know it. Okay. And this is a paper from photo energy. So where you see in the area of providing a solution to energy problem. So dioxide solar cell. So I'm just giving you an idea how you can explore this particular IR spectroscopy to provide a strong evidence in favor of your hypothesis of your research problem. So see what is being done in this paper is they have extracted a natural dye from wormwood and a purple cabbage, and that dye is actually mm, that dye is actually uh, uh, it's found to be an anthocyanin dye, and they have a target to use this uh, in the licensed solar cell. So basically, the idea is the comparison of an IR spectrum of chlorophyll with the dye. So chlorophyll, you know, chlorophyll is a photosensitized material which is responsible for photosynthesis. So it is absorbing the light and converting it into other form of the energy. The same way, the dye they extracted, what is the hypothesis this group of people they have proposed? That it is almost showing the same peaks which were there in the chlorophyll. So just to provide an evidence in favor of their hypothesis that whatever the dye, natural dye they have extracted from wormwood and the purple cabbage, that shows a similar peaks like this peak, this peak. So these are the similar peaks, right? 1, 2, and 3 peaks, right? So these are the 3 peaks which are actually comparable with that of chlorophyll, and this supports their hypothesis. This is just an example to tell you how you can find out a scope to utilize the IR spectroscopy for in your research area. One more example that I would like to tell you is about the study of silver nanoparticle hemoglobin interaction and composite formation. This was a paper published in Colloid Surface Interface by Threads and Surface Biointerface. So what they did actually, they noticed that this is actually a comparison of an IR spectrum between the pure HB and HB with silver nanoparticle. So definitely this interaction will be definitely helpful to provide a solution to many diseases and many problems. So to understanding the interaction between these two, they used IR spectroscopy. In fact, not only IR, but there are other techniques as well. But as I mentioned, no spectral technique is perfect in its own. You need to use all the spectral techniques together, and you need to identify that you should have some thought process behind the use of this any spectral technique in your research problem. So here what is happening, the silver nanoparticle can attach and come closer to the heme, and they also found that there occurs a conformational change and the hemoglobin HB becomes unfolded through the increment of the beta sheet structure. There occurs a change in the structure of hemoglobin when the silver nanoparticle interacts with them. This was the conclusion they could draw from the IR spectrum. Of course, they have used other techniques as well. This is just an example. Whatever may be your research area, you can always explore a possibility to use IR spectroscopy for your for proving your hypothesis. And this is an example from the forensic science. So biological stains from forensic purpose. So you know when you take the blood, so this is actually a data that you can get from this journal. So you see in the blood you get a peak at 3292, 2956. So you all these peaks have been assigned. The normal assignment that we have seen so far. Okay, so the normal assignments that you so whatever samples they have collected, say blood, saliva, they have collected, and in the forensic investigation, they have collected that and they are using that for analyzing the presence of these particular compounds in the in that forensic sample. So blood and saliva they analyzed and they found that these are the peaks. So whatever the sample they collect during that and they analyze with it and they found and they correlate with the that is going to help them for the forensic purposes. So these are some of the examples that I have tried to share with you. So this is all about the IR spectroscopy. Another 10 minutes or so, I will be talking about the research papers. Can I, sir? Rasade, sir?
Yes, sir. Sandesh, sir.
Yes, sir. Yes, sir.
Yes.
Okay. Okay. Power, sir, is it going well?
Yes, well.
Okay. So now as a researcher, we do research. And what normally happens is most of the teacher fraternity, maybe you are the people not doing that, but most of the teacher fraternity, they are publishing their papers in the fake and the fraudulent journals, the fake journals with the fake impact factor and the fraudulent journal. Now, it is really, and I take this opportunity to to propagate my thoughts on this particular topic, that where and how to publish. The first thing first is we should do a good research, a good science has to come out. You can do. So I have a strong collaboration from Daniel Seidel from USA, IICT Hyderabad. So when you have a strong collaborator, you can come together and you can definitely contribute a good science. So by any means, you should produce a good science. And if you are doing good science, you should publish in good journal because it should reach to the good paper. Because the paper, when you publish, if you are doing a very good, very hard work and you are doing a good science and you are publishing it in a fake and a fraudulent journal just because you are unavailable journal, then I think you are committing a crime. So you are guilty and you are committing a crime. So that should not happen. And for that purpose, I would just like to tell you that, what about the quality of the research journal? Now, government also wants, see, in the context of the global rank, India is lagging much behind. So when you look at the first global ranking, you will get the first global for IIT Bombay in the QS ranking. So you mean that across the globe, there are 171 institutions which are ahead than our topmost institutions in India. And one of the reasons is quality research output. And see, government has done so many tricks for improving the quality of the journal. So they came up with API in 2009, did 4 amendments in that. Because, I'm very sorry to say, but our teacher fraternity, especially in higher education in the country, they are always engaged in some sort of jugaad, what you call. Because you see, if you see the number of conferences and the participation of teachers, if you see the number of conferences and participation of the teachers during the 2011 to 2014, you will find that most of the teachers, most of the colleges, they have organized conferences and most of the teachers, they have presented their paper, because all of a sudden in the first API UGC regulation, and it has given a huge weightage to the people attending conferences and presenting the paper. So later on, they realized that people are doing this kind of burglary. So what they did amendment and they have reduced the weightage for the conferences. And now you see, it is very difficult to organize a conference and getting participation of actual participants in the conferences. If I'm not wrong, this is a very common situation all across the country. Then what happened? Later on, they came up with the UGC approved list and they published it on the UGC website. And later on, they found that UGC approved list was again prepared by the Board of Studies of the affiliating university. And Board of Studies have the teachers. So again, it is in the hands of the teacher to save the teachers for the sake of their career advancement scheme. So what happened? There were many fake and fraudulent journals in the UGC published. So from 14th of June 2019, they came up with UGC Care. So now the the journals which are part of UGC Care, they are supposed to be the approved. They are supposed to be a relatively good journal, and unfortunately, our country has become a hub for the predatory journal. So they want you to work hard, they want you to do science, and they want you to they take money from you to publish your work. It means you are creating a new knowledge and you are paying money to make it available in the public domain. So this is what is happening actually. This is all the concept of predatory journal, and unfortunately, India has become an hub for the predatory journal. Every gully mohalla, one day a journal, if UGC Care takes an action against one journal, next journal comes from the same mohalla next day. So likewise, you are always being thrown the predatory publishers, predatory publishers who will ask you that, they will claim that we are part of that, but they are not so. So always follow UGC Care guidelines while publishing. And I would suggest that there are two groups. When you log in on the UGC Care Pune website, you will find that there are two groups, and I think one of the resource person will be talking about this in more detail. So I will not talk much, but I would just make an humble submission that this group 2 involves a globally recognized database. So I request all the participants, whatever the science you do, try to find a journal from the globally recognized database, because group 1 list is very, very dynamic. Journal which is in a part of a group 1 today may not be a part of a group 1 tomorrow, because the list is really, really dynamic. Okay. Now, when it comes to the impact factor, there are about 1000 methods of calculating impact factor. There are 1000 agencies, they have developed their own methods of calculating the impact factor. But these impact factors are not valid. Only the impact factor which is published by the journal citation report by Clarivate Analytics, which was earlier, it was Thomson Reuters, it is only considered. And how is this impact factor calculated? Just to give you an overview, it is based on the performance of a journal for the 2 consecutive years. So, and it is simply a ratio of number of citations divided by number of articles in the 2 year. And when it comes to because being an accuracy coordinator of my college, I have to screen the CAS proposal. So many people came up with, say, they claim that our journal is having an impact factor of 6.5, and then when we say, no, yaar, this is not, this journal is not a part of this list. He said, no, journal is claiming that it is having this much impact factor. So all such impact factors are not being considered even for crash commotion or even for any other. This is just to, misguide you to publish your good science or good work in the fake and fraudulent journal. Then when it comes to the cash promotion, normally there is some what we call unwritten rule that you should have this much citation, this much H index and this much I index when you are going for the promotion for associate professor or professor in the college or in the university. So one should be aware about the bibliometrics of research publication. So what are the bibliometrics? Number one is citation. The number of times your work is cited in the paper. H index is the number of papers with the citation number higher or equal to H. I will explain this in a very simple way. Say I am an author of 8 papers. I have arranged these 8 papers in the decreasing. So first paper got 21 citations, the last paper got 3 citations. So I have arranged it in the order of decreasing citation. Okay. Now, what is my citation index? The citation index is the sum of all citations which I have received for all my articles. Then what is I10 index? I10 index is the number of papers which have received 10 or more citation. 10 or more. So 6 number of papers are there which have got serial number 6. Up to serial number 6, there are 10 or more citations. So I10 index is 10. And when it comes to H index, you see there are 7 number of papers which have got 7 or more citation. So 7 number of papers which have got 7 or more citations. So it is H index is 7. Now if I have got 6 citation for this paper, then my H index will reduce to 6, because there are 6 papers which have got 6 or more citation. In order to transform the H index from 6 to 7, I need to get one more citation for my 7th paper. So why these bibliometrics are there? This is just like to check the contribution of a researcher and consistency of publication. Consistency is one parameter. You know, we often watch cricket, and you know there are players like Lokesh Rahul. He scored 300 in a test match, right? Lokesh Rahul at one time has scored 300 in a test match. But if you see the people like Virat Kohli or Rohit Sharma, they are consistently performing every now and then. They are bringing their 50s and 100s and they're contributing to the. So overall contribution is measured through this I10 index and H index. So these are the bibliometrics that will give you an idea about the citation. Because I have seen one or two authors, they did say like when they do their PhD and when they do their postdoc, they got one very fantastic paper. So I know a person whose overall citation index is 1000, and out of which only one paper has got 700 citation. Sorry, 700 citation. What does it mean? He has done really good. One paper he has done really good, in which for which he got 700 citation. And for remaining of the say 20-30 paper, he has got only 300 citation. So to check the consistency, to check the consistency, it is very important that, because normally you can publish a paper, say if you have joined some very good group for your postdoc and you have got a very big paper, when you may get a very good number of citation for that paper. But when you come back to your place, you join somewhere as an assistant professor and you're not doing research for one or other reason, then your bibliometrics will reflect that your performance. So bibliometrics are made for that. And the last point that I would like to talk about is about creation of the profile. So our visibility as a researcher, in the on the web is really important. And for that sake, I would say that we should start creating our research profile. One of the simplest kind of a research profile is a Google Scholar, which you can easily prepare by using your Gmail account. Other is by Scopus. You should have your Scopus account. You should have an ORCID account. ORCID profile. LinkedIn, these are also there. You can have the Publons. So when you have a Scopus, you got Scopus. Normally, whenever you have an publication in elsewhere, or you have done some review in elsewhere, you probably can have the Scopus, or not. Even if not, you can go to the Scopus site and you can do that. And when it comes to the Web of Science, you can have the Publons. And Vidwan is a similar platform which is started by the IncludeNet.
Okay, sir.
Okay. Okay.
Wonderful, sir.
Okay. Last point. So these are some of the points that I would like to tell you. Have your profile. So IR alone cannot determine the structure, but functional group you can easily determine. And sometimes negative inference is very powerful. Absence of a signal for a definite proof is a definite proof that functional group is absent. And you can always identify, I request all of you to identify a suitable application of IR in your research area. And I would say do good and publish good science. That's it from my end. Thank you.
Okay. Thank you, Sandak sir, accepting our invitation in 11th hour and prepare this topic on the very important concept regarding the IR spectroscopy. Okay, though I request for participant ask your doubts or query. 1 or 2 query are accepted. Any query or doubt from the participant regarding this IR spectroscopy? Unmute your mic and then ask.
Sir, we have seen the use of probes to follow the reaction. So, Can we analyze transient species or short-lived species which are formed during the reaction using this probe?
I think we can, because probe is means in real, you are doing a real-time analysis. In the flask you have a probe. So I think you can do that.
Because the transient species are very short, they are very short-lived. For a few seconds they appear.
Yeah, but you can do it like the way we do the time-dependent NMR, wherein we put the sample and you take a running NMR there with time. So you have to record. You have to basically you should know when to record your spectrum. You need to know the frequency of recording the spectrum. Then probably you can definitely do that.
Okay, thank you, sir.
Okay, now I request Mujab Sheikh, please give.
Good morning. On behalf of Ramkrishna, Professor Ramkrishna Murthy of Jagpur and HISP Pune University, and on behalf of all the participants, I take this opportunity to thank Professor Hemant Kumar S. Chandak sir from GS College, Khangaon. Sir's topic usually spectroscopic analysis of chemical and biological samples, and our FDP's topic techniques for research in clinical and biological sciences. So the topics itself explains the aptness of it, what it was all about. Sir started this lecture by explaining the importance of having the knowledge of a structural knowledge of a particular compound, so as to benefit it to both the group of chemistry and biological sciences. He has given a good example of cholesterol. How from the history itself, he tries to explain that one day how long it takes to understand the structure of cholesterol, and how it is now necessary to understand the structure, and what one can do by knowing all these things. So sir started from the basic fundamental structure of fundamental properties of IR spectroscopy. Then he explained the main IR. Then he proceed towards how to handle the how to have the techniques for handling the sampling techniques. And then later on he explains the vibration frequencies with the example of H2 and I2 examples, and how the molecular difference can fluctuate the vibration frequency. So later on, sir also explains how to interpret the spectrum that we get it from the higher spectroscopy. Ultimately, to me, what was very much important, as I am a biological background as a student, so I actually was very much interested to know how actually this spectroscopy is working exactly, and what we can do. Although we have known all these biological compounds, but how exactly what these functional groups are and what they are working. So this all curiosity of mine, and I hope all of our participants has been clear through this wonderful lecture of sir. Sir also explains the application of IR spectroscope in a very beautiful manner. Overall, I can say the sir's lecture was overall complete package, from knowing the functional group, knowing the structure, then functionality and its application, till the publication. So it was a kind of complete package. At the end, I thank you, sir. Thanks for delivering a wonderful lecture. Thank you very much.
Thank you, Mujab, to appreciate our resource person to delivering the talk on IR spectroscopy, and also thanks to Dr. Hemant Chandak on behalf of our institute and our college and all science departments, and also from the HRDC. Thank you, Chandak sir, to accepting our invitation and deliver the speech in our FDP. Okay, thanks again. Now I request to Varsha Sriram, please take the charge of the next session.
Okay.
Sriram, madam.
Yes, sir.
Yes. Chandak sir, we can. Okay.
Thank you, sir. Thank you so much. I am here to introduce our next speaker. Uh, yeah, Dr. Vittal Karakar, sir. Sir is assistant professor in the Department of Botany, Savitribai Phule, Pune University. Sir has completed his BSc, MSc, PhD in the subject of biotechnology. He worked in National Chemical Laboratory, Pune for his PhD. He also visited Israel in the tenure of his research. His research areas are plant genome, and multigene family evolution, abiotic stress tolerance, and secondary metabolite synthesis. He received the Best Research Scholar when he was working for his PhD. Sir also has awarded the Best Published Research Paper in Biological Science in NCL. He successfully completed his 5 research projects, and after his PhD and as an assistant professor, Sir has published 21 papers in peer-reviewed with repute and high impact factor journals. There are 6 book chapters on his credit. He has 226 citations on his credit. His H index is 7 and I-10 index is 7. And the really appreciable thing is he could achieve all these, that is academic as well as research achievements, and and his publications as well. All these credentials he could achieve in a very short span. He's so young, and he has long way to go. So I wish him all the best, and I request sir to deliver the lecture. Thank you so much.
Yeah, thank you for kind introduction, Dr. Sriram. So it is really a pleasure to be here. First, I would like to thank organizing committee and Dr. Sriram for inviting me to deliver this lecture. So as I understood from the organizers that the participants are mixed from chemistry and life sciences, and I will try to give justice to both of the streams. And you agree with me that most of the chemistry Nobel Prize are won by the biologists, and that's how the interdisciplinary work is important, and it has really uh, immense applications as well. Okay, so before we start, I'd like to propose few things. I have a long slide list, but it's not completely necessary to go through all the slides. And I, since as Dr. Varsha mentioned that I'm very young and I have also been going through this refresher programs, and I know the pain of attending these. Until and unless you give some interesting talks, most of the participants are not at all interested. So I know the things from other side as well. So as I mentioned, it is not necessary to completely go through all the slides. I will try to make the lecture interesting, as well as whenever you have any doubts, or if you want to emphasize on a particular point, we can stop at that point and discuss in detail. Ultimate aim of this lecture is to understand something about the transcriptome technologies that are being used recently very widely, and if some of you take these technologies to your research and practice it, that will be a major success of this lecture. With this, I'll share my screen if it is allowed. Is my slide visible? Dr. Yeah. So the title I have chosen for the speech is Plant Omics and Their Applications, Riding the Data Science Wave. I have specifically selected this catchy title because all of you might have heard the word data science and the analyzing data. So next a decade would be revolutionized by the data science, and that data can be anything, even uh, the everyday's WhatsApp post or Facebook post you are posting on any social media can also be considered as a data, and that also people are analyzing and commercializing. So you will be surprised that there are many startups which are using these kind of data to give you customized and personalized advertise, and depending upon those advertise, they are earning a lot of uh, grants as well as funding. So data, that can be anything. So since we are working in life sciences in terms of a life scientist, for us data is anything, like it can be a list of metabolite, list of genes, sequences of genes, the whole of genome sequence, and anything can be considered as data. And analyzing and acquiring that data is huge field, and people are working in these areas to understand various aspects of biology, not only biology, chemistry, biology, and environmental sciences. And we try to touch upon various aspects of different omics. So if you see, there are omics is nothing but a technique which can parallelly at high throughput scale analyze any biomolecule. It can be a metabolite, it can be a transcript, it can be a protein, it can be a whole genome. So if you are able to analyze and sequence multiple transcripts, it is named as transcriptome analysis. If you are analyzing multiple protein at a time, and identification and quantification, then it is proteomics. Similarly, Paul, if you are identifying and quantifying multiple metabolite in a cell or in a tissue under different environmental or different conditions, then it is a metabolome. So for each of these techniques, omics technology, there are different tools available. And for example, for transcriptome analysis, next generation sequencing is there. In today's lecture, I am only focusing on transcriptome. Although I am working on all these aspects, but since it would be because of the time constraint, we can only focus on one of the technologies, and more or less rest of the technologies are similar. Only way of data acquisition is different. In case of proteome analysis, you need a high resolution mass spectrometer, which can fragment all the peptides and analyze the concentration and identity of peptide and proteome, and ultimately will get a concentration of individual proteins in different treatment conditions, tissue or whatever experimental conditions. Similarly, you have metabolome analysis. So we generally work at metabolome and transcriptome level for understanding different pathways. Okay, so I'll be concentrating only on transcriptome analysis and how the people are using different next generation sequencing platforms to acquire nucleotide sequence information, maybe genome sequence information or transcriptome sequence information. So if you look at the history of DNA sequencing, for those who don't know what is DNA, DNA is deoxyribonucleic acids, and which is an information storing molecule which is there in each and every living cells, and all the information is stored in DNA, which will execute through transcription, translation, and the phenotype of an organism or a cell can be decided. So the genetic material structure, that is DNA structure, was established by Frederick Crick and James Watson in 1953. For this discovery, they got also Nobel Prize. And then after the discovery of DNA, that is DNA is a genetic material that was established. And then obviously DNA consists of only four types of nucleotide, but their arrangement across the length is which ultimately gives rise to phenotype. So, sequencing of those the array of ATGs is important, and this method is known as DNA sequencing. Understanding the sequence of these ATGs in along the length of the DNA or transcript RNA is possible nowadays because of the next generation sequencing. So if you see the timeline, how the sequencing methods have developed, a lot has happened over the years. But in the recent decade, like from say 2010 to 2011 to 2021, there is enormous things have happened in terms of sequencing of the nucleic acids. And now we are in era of third generation sequencing, wherein the whole genome of human can be sequenced in a single day. So the instruments have evolved in such a way that you can sequence whole genome of human. So earlier it took around 20 years to publish or to come up with the complete human genome, and that was the milestone project for whole of the humanity, where you could identify a sequence of each nucleotide in your genome. And now at today's date, you can sequence whole genome or even whole plant genome in a one day, because of the advancement in next generation sequencing platforms. And I'll briefly go through what all these next generation sequencing platforms are there and how people are using them. So if you see how the DNA sequencing technology has evolved, so initially it was Sanger based and having manual slab gel where throughput was very, very less. In one run, you can get only 100 base pair sequence. But with the replacement of fluorescent dyes, again, the credit goes to the chemistry people who come with the nucleotides which are fluorescently labeled. And in automated slab system, also we call it as a Sanger sequencing or normal capillary sequencing, wherein the read length was increased to say 700 base pairs to 1 kb, and then people actually parallelized it, having 96 well plate, and you get 96 sequences of say around 1 kb in one run. So, but then the things have changed very drastically rapidly after 2005, wherein a second generation capillary sequencer and the next generation sequencing platforms came up, which gives short read sequences, and but the amount of sequence information they were giving was huge in terms of millions of short sequences. Okay, so in coming years, you can see the sequencing cost has also reduced. Right from earlier when the human genome project cost was in billion, but right now in one day and in few dollars also, you can sequence the whole genome. So cost of the human genome has reduced drastically. In fact, nowadays in $1,000 you can sequence whole of the genome of human, and it is because of the advancement in sequencing platforms. Okay. So as I was mentioning about the data, so if you compare the amount of data coming from all the social networking sites, maybe Twitter, Facebook, or any other social networking platform, if you compare all the high resolution photos people are posting on say, Twitter and Facebook, it is less than the amount of data generated by life scientists, and this is surprising, isn't it? So nowadays the amount of data in terms of GB, gigabyte, it is more for generated by the life scientist, and it is mostly either gene sequencing data or transcriptome sequencing data. There are many ways you can generate a nucleotide sequence data, and all of this data, all of this next generation sequencing data is freely available, which is surprising, isn't it? People invest a lot of grants in acquiring the data, but whenever they publish, it is mandatory to submit all their data into a sequence data archive at the NCBI database. So if you just sequence data, and if you have a specific paper in which you want to download the data, it must be available at SRA. So those faculty or teachers who don't have a lot of resources available or don't have grants, then they can use this data, analyze that data. And even in my lab also we try to do this kind of download the data and ask different questions were not being asked by the original analyze data and publish. So we have quite good publications by analyzing other data as well. So what you need is just a computer and knowledge of how you analyze, how to make a sense out of these huge data available in the public domain. Okay. So now it is kind of a cartoon wherein the limonite is costly than that of the whole genome, and this is because of the there is wide upliftment of these platforms, and now the genome sequencing is very easy and cheaper. But this has created another problem. Now, every now and then, people are giving their bacterial samples, genome sequencing or transcriptome samples, but once they get the data, they get drowned in the next generation sequencing data because you have huge amount of data and you are now drowned, or you are now piled in the data, and you are not able to draw a meaningful conclusion. This happens because there are multiple reasons. If your experimental design is wrong, if you are the way of sampling is wrong, the way of platform on which the data was analyzed is wrong. So whenever you plan an experiment for next generation sequencing, you need following things in mind. So we discuss those things. Okay, so there are NGS machines available, and because of cheap nature of sequencing, the huge massive amount of sequencing data is there, and now PhD students are asking, what should I do now? We have so much of data, how to proceed? Okay, so before we go into the application part, I would like to briefly go to the different next generation sequencing platforms which are currently in market. So most of the major market is dominated by a company known as Illumina. The Illumina has a long history, but currently it is the major platform that people are using for generating genome sequence data or transcriptome sequence data. There are various machines, NovaSeq, HiSeq, NextSeq, MiniSeq, MySeq, iSeq, and all these are different versions, and each of these versions has its specification and properties in terms of read length, the depth and amount of data generated, and so on. So in case of Illumina, what you start with, you isolate good quality genomic DNA, and then you can switch to any sequencing company. What they do is they take the genomic DNA, fragment it by sonication or by using some enzymes such as restriction enzymes, and then they live these adapters, adapter A and B, also known as sequencing adapter. And using a bridge PCR, they generate DNA clusters like this. And the Illumina platform works on reversible terminator dyes. Okay. Reversible terminator dyes. So each of these nucleotide ATGC, they are being attached with some kind of a fluorescent dye having different wavelength for detection. Okay, so one adenine will fluoresce say red, one will fluoresce green, and likewise. So and DNA polymerase is added here, and DNA polymerase will add only one base. And because the 3' end is capped, so it cannot add the next corresponding complementary nucleotide until that terminator dye is removed. And during removal, it will absorb or it will capture light or fluorescence like this. And in each cycle, you will have fluorescent tiles, and the computer will convert these fluorescence into a sequence. You can imagine these each DNA cluster will have a unique sequence. There will be millions or millions of such clusters on a small plate, like say 2 centimeter, 4 centimeter plate like this. And at the end, you will have millions of such short reads, and that is your raw data. And then that can be assembled and analyzed further to come up with the whole genomes. What are the advantage of these platform? Illumina, it is having high read accuracy. This is one of the platforms which is having high read accuracy. That means whatever nitrogen bases or nucleotides they are being read or sequence is being read is highly accurate. Okay. Then the amount of data generated is very high. The shortfalls are high platform cost. Even till today, these platforms are really costly. So mostly it is advisable to outsource this. So there are many institutes who have procured these platforms, but since the amount of investment required to run these platforms is very high, as well as the upgradation in these platforms is very quick. So once you buy, say, NovaSeq, in 1 year that will be absolute and new platform will launch. And on top of that, we stop giving chemical consumables to the old platforms, and that will be a kind of white elephant. So it is always advisable to give your samples to commercial vendors. In less cost, they will also give good data. So this is the major platform which is used for next generation sequencing. Second platform, Thermo Fisher scientific platform. Even this year, this has been obsolete, or people are not using this because of inherent issues with the this iron torrent platform, or they also call it as proton personal genome machine. When it was launched, the idea of Thermo Fisher, initially it was launched by Type Technologies. They call it as a personal genome machine, and their aim was to each and every individual will diagnostic centers will use this and sequence the patient's genome, and then they find out the single nucleotide polymorphism, and depending upon that, the doctor will prescribe. But there not fulfilled because of inherent issues with this. So here also you start with the total implementation to immersion PCR, and in this case, instead of fluorescent table, it is the principle is based on release of hydrogen ion as soon as nucleotides are being added. So this is also known as semiconductor sequencing because uh, the plate has an ion sensitive layer. So whenever nucleotide is added, a proton is released, and that release in proton changes minute pH. That change in pH is monitored. So the very accurate and sensitive pH meters, very small pH meter, are at the base of these plates. And if a nucleotide, then proton will be released, and that small change in the pH can be recorded, and it will give a sequence like this. So the read accuracy is less in the region of polymers. The read length is around 400 base pair and shorter runtime. These are few pros and cons of these iron torrent or Thermo Fisher scientific platform. Okay, even this platform is also obsolete now. Only Illumina and the later two platforms, that is Pacific Bioscience platform, PacBio in short it is known as. So this is the platform which gives a read length of around 10 kb or more than that. So this is also known as single real-time sequencer. So this was possible because of advancement in chemistry as well as biochemistry. So you will appreciate that this was possible because chemists identify a enzyme which will be immobilized into wells like this. Okay. And this enzyme has an ability to pass a single nucleotide strand like this. This is the polymerase again, and this will add nucleotides, next complementary nucleotides here. And the nucleotides, they are fluorescently labeled like this. And as soon as new nucleotide is added, the fluorescent tag is removed, and that will give a short-lived fluorescence and will be measured. So this is also known as single molecule real-time sequencer because only one molecule can be passed through this polymerase, and you will have very long read length. And most of the genome sequencing project now people are using this, but the problem with this is there are more chances of errors in the reading, or errors created by DNA polymerase, and so on. So very high platform cost, it has lower throughput, low read accuracy. The only advantage of this platform is a very long read length. So if you compare the read length of Illumina, which is at the max it is better, but in case of platform equal RS2, it is around 60 kb, which is very big. In 4 read to have whole equalized genome, which is very nice. So people are now using bio and as a hybrid to come up with the genome or transcriptome as well. The last platform is very interesting platform, Oxford technologies. And instead of sequencer, you can imagine this is a like a palm sequencer. In your palm also you can fit the whole sequencer. There are multiple versions of it. This is again a single molecule sequencer, and this PacBio and a nanopore sequencer as third generation sequencer because the read length is very. So how it works? Here again, chemistry is a lot of chemists have invested their resources and time to come up with these nanopore technologies. So in this, sequencer, you have a lipid bilayer which is electrically resistant, and there are proteins as SPP, where it's a transmembrane protein and which has pore, and that's why they are being categorized. And there is one more protein as 5 DNA polymerase, which is from Bacillus subtilis bacterial 9. And the DNA polymerase, very usual DNA polymerase, which takes the and displaces one strand and pass that strand to this protein. And these pores are so narrow that can be passed to the stranded DNA. And this single stranded DNA, it passes, the it has different eigen bases. So what is measured is change in the layers. Since is electrically resistant, so the voltage across this lipid barrier is measured. So as soon as individual nucleotides are passing, the cell and DNA passing, and nitrogen bases will change current through these pores, and that is dependent on type of nucleotide. So if it is passing, change in current is X. If it is passing, change in current is Y. And you can measure which nucleotide will pass to the pore, and you can imagine how precisely people have built it. And this is a major sequencer which has been used in COVID pandemic. Most of the SARS-CoV-2 genomes have been sequenced by nanopore, and it was possible because the nanopore was there to sequence the whole genome very rapidly. Within a day or so, you can sequence multiple samples of SARS-CoV-2 viruses, and you can align them to the original strain and come up with whether there are variations. And now news you might have heard of different variants of SARS-CoV-2, and those have been identified by this technology, Oxford Nanopore. And people have been sequencing the SARS-CoV-2 genome worldwide, and there are repositories where they deposit and analyze whether there are new variants or the same variants are being propagated or creating a pandemic. Okay. So the major issues with this is the low platform, like reduced library representation, and then it is also portable. So portability is one of the major advantages of nanopore technology. You might have heard in news that these are the sequencer which will be fit in the astronauts which are going in other space, where if they find some genetic material there, they can sequence the nanopore technology. So in next missions might be we will have these DNA sequencers on the rockets or launchers as well. Okay. This is a summary slide wherein you have comparison of multiple platforms. So these are the internet sequencer. This is thermometer, these are PacBio and nanopore sequencer. So currently, these two systems are very widely. So the latest one is NOAA 6000, which has a capacity to generate a short read of 150 base pairs and paired ends. So I'll discuss what is paired end. And then you can see the cost, mostly paired end reads. Apart from that, increasing the read length beyond 150 base pair in case of human, there is no improvement in the full length reconstruction of transcripts. Okay. So if you are working on plant or say any animal system, so 150 or 200 base pair read length is more than sufficient. Don't go beyond that. Okay. So the second question is, should I acquire data in single end or paired end mode? So Illumina gives this facility of paired end reads, wherein the genome is fragmented, and then the ends from both the sides are read or sequenced, and having in between the insert, they call it as the insert. So apart from sequence information, the paired end reads also gives them positional information. That means the insert size is known most of the time, it's around 300 to 400, and if it is known, then if one sequence is aligned to the genome at X position, then the second paired end read should be aligned after 350 base pair. So likewise, this kind of information is also useful in the assembly. Okay. So this is helpful. Like paired end data is helpful in resolving the structural rearrangements such as deletion, insertion, inversion, and so on. So this also helps in differential gene expression analysis, and obviously the paired end data is more costly than that of the single end data. Okay, so if you have a grant and if your project allows, then always go for the paired end sequencing and not for the single end sequencing. Okay. So while paired end data runs are costly and time consuming, you get twice the data, twice amount of data in case of paired end than that of the single end. Okay. So, but if you have a known sequence and you just want to count the expression, then even single end reads are also fine because they align faster. The acquisition is also cheaper, and the single end data is also sufficient for just doing RNA sequencing or understanding the profile of transcriptome. Okay. So next question is whether to go ahead with the technical and biological replication. Okay. So you might be knowing that for any experimental design, you need to have a technical as well as biological replicate to give your data statistical significance. Okay. So in case of Illumina, the Illumina platform has very low technical variations, unlike microarray. Hence, technical replicates are not necessary. Obviously, all these technical and biological replicates will add cost to your whole of the project. So it is advisable to not to use technical replicate because run-to-run variations are very less in case of Illumina. And to avoid a batch effect, try to sequence everything for a given experiment at the same time. But it is always avoidable to always advisable to sequence everything in same cell, flow cell or batch, just to avoid any batch to batch variations. Okay, right from library preparation and sequence acquisition, you can do in the same batch. There is no need of technical replicate, but always keep biological replicates. So minimum 3 biological replicates are must for sequencing. Obviously, this will add cost to your whole project, but if you want to publish in good journal, always they will ask for this biological replication, and until unless you give some statistical state and significance, then only the reviewer and the editor will believe that these genes are differentially expressed. Okay, so RNA sequence differential expression studies, more you want more replication. So this is a paper which says that the more or less 3 biological replicates or more the replicates you get, and instead of sequencing data or have more replicates, the power to your differential data, where do I start? So obviously, this is the major question all the biologists are facing, because sequencing is not a problem now. What you have to give is just either give the whole tissue to the vendor or isolated RNA or DNA to the vendor, and they will give you raw data or analyzed data. But what do I do with that data? That is the major question. So first thing, whenever you get a raw data of the sequence information, check the quality and desirable or expectations from your data. The first thing you need to check in your data file is data amount. Say many times these vendors give tell you that I will give you 5 GB data. So then first find out that whether the amount of data is as per our expectation, number of reads are as per our expectation. Next thing you want to check is data quality, whether the supplied data or given data is a good quality data. Good quality means, say these are the 2 pictures of FastQC output, wherein the read length is 50 nucleotides, and on x-axis you have nucleotide position 1, 2, 3, 4, 5, and on y-axis it is quality score or Q score. I'll explain what is Q score, but it is a tendency of Illumina to get the quality gets deteriorated as the read length increases. So you will get a little bit decrease in the quality, but it should not be like this. Okay. So what does this Q score indicate? Q score indicate is what is the probability of accurate calling of this position of individual bases. Okay, and you need to ensure that whatever raw data you have got from the vendors, it is of good quality. And there are many tools available to check the quality of data, FastQC, and there are NGS QC, Trimotic. There are many number of tools. All of them are good. You need to check whether the quality of my data is good or bad. So most of these files, these files are in GBs. If you try to open it in your Windows computer, the first thing that happens is whole of your laptop or computer will hang because you cannot open bulky file. So you need to have understanding of the Linux database or a Linux operating system to open and see those files. So if you open few reads from those files, each of these reads will have 4 lines. The first line is the header line, second line is actual sequence, third line is plus sign just to separate the quality score of these sequence, and the last line is a quality score or Q scores. Okay, in the same header line, first line you have instrument ID, flow cell ID, and all the information. So why I'm telling you that whenever you get the data, see to that all the reads are coming from the same instrument. Many a times it may also happen that vendor will pull any random data from SRA, they will download and give it to you. You need to understand whether the data is acquired on the same machine or with the same flow cell ID, so that whatever you are analyzing makes some sense. Okay, so how this trade quality score is calculated? So these are the scores, although you see here some characters, but they have been coded, and the coding is in ASCII 2 characters. So say for example, quality score is 10, that means there is a probability of 1 error in 10 bases. If quality score is 30, that means there is a probability that there is 1 error in 1 KB stage. Okay, so most of the times scientists prefer trade quality score more than 30. Okay, and there are tools available to analyze quality of each of these base, and if you do the fast QC check, then it will give you the quality of each of these. And this is the ASCII coding. Say for example, this is the base and corresponding quality score is G here, and this is the ASCII table or coding wherein G corresponds to 39. Okay, like this here. This is how they calculate. 39 means it is more than 30. That means the probability of the red A or the base which is called as A is wrong or which is correct is almost 99.99%. Okay, so which is quite a good quality, and most of the time even for Sanger sequencing we get a trade quality score. So quality is very important, because if there are some errors and if you try to align them, and if it aligns to the genome and it shows some error at particular position, you might assign that error as a single nucleotide polymorphism, but which is not, because it is a error in the sequencing. So if the quality is good and then still it is mismatching with the genome, then you can assign it as a single nucleotide polymorphism. Okay. Okay. So once you analyze the whole quality, then next step is doing the differential gene expression analysis. So you have a lot of different data sets and different platforms. The only bottleneck here is bioinformatics or analyzing the data. Okay, so whole of this huge amount of data can be analyzed, and but you need good hands-on on a bioinformatics. You need to know all the softwares, and there are many available pipelines on the internet, or people have this. You need to understand them and analyze your own data. But till today, the only bottleneck in analyzing whole of this data is bioinformatics, computational power. So all this high throughput data can be analyzed by various tools. You can first collect the data, then reduce the data, remove the redundant, do the variant calling, map those short reads to the genome, and then identify number of reads which are aligned to the genome or transcripts, and then do the gene expression analysis, differential gene expression analysis. Ultimately, all those differentially gene expressed genes can be assigned to a particular pathways, or you can understand the ontologies, or you can build a network to understand what is exactly happening in a particular control treated condition. Okay. So this was brief background about the next generation sequencing. With this background, we will try to understand what all applications these sequencing platforms have in plant sciences, and this is also true for any other biological science. I have just taken plant as an example because we work in plant system. So you can do a transcriptome sequencing, exome sequencing, only the express part, allele mining, metagenomics. Metagenomics is very interesting field wherein all those organisms which can't be cultured, and if any of you are from microbiology background, you will agree with me that only 1% of the microbes are now cultured, and more than 99% of the microbes still cannot be cultured because you don't know the media, you don't know the specific condition to grow them. But metagenomics is a field wherein you can't grow them, but you can sequence them by using next generation sequencing. Say people do extract the whole of genomic DNA from the soil, and then they sequence and identify what all culturable and non-culturable microorganisms are there in association with the rhizospheric soils or any other. Even human gut microbiota is also a very nice example of metagenomics. Okay, so single cell genomics, then phylogenetics and ecological studies, epigenetics. So all sorts of these applications can be done by using next generation sequencing. And few of these applications I'll emphasize and go into the details in coming slides. Okay, the first application, which is whole genome sequencing for plants, I have mentioned here. That is also true for many mammals or even many bacteria as well. So till today, more than hundreds of the plant whole genome have been sequenced. And if you visit a PLABI database, you will see there are lots of different crop plants and the model plants they have been sequenced, and whose genome sequences are freely available. So if you are really interested in understanding the genome, you can get the genome sequence information of the cost at many websites or web servers. A few of these web servers are, say, Phytosome, Ensemble Plant, Plant GDP. All these are common web server for accessing and analyzing the plant genome database. People have also generated a specific databases, genome databases such as Grameen or legume information system. So this is for leguminaceae, soybean database for soybean genome. Then you have TIR for which is the Arabidopsis information resource. So wherein all the variants of Arabidopsis thaliana is one of the model plant, and a lot of things have been done in Arabidopsis, and whose genome sequence information is available along with lot of other data, and which is freely available. So if you really want to work on any of the plant whose genome is sequenced, only a whole genome is at your fingertip. You can just Google or visit any of the database, isolate gene of your interest, look for any variations, do the phylogenomic analysis, and so on. Okay, so this is a major application, that is whole genome sequencing of any life forms. Okay, then this is just for an example I have given. So I have been as an genome database here at Phytosome, and you can visualize specifically chromosomes, zoom into a specific region of the chromosome, identify what it codes for, analyze the protein functions, and then you can isolate, make a synthesized primer to isolate that gene of interest, and many things can be done using this database. Okay, so second application is genotyping by sequencing or genome-wide association study. In short, it is also known as GWAS. It is exactly different than that of the normal linkage map, wherein you do recombinant lines and then construct a linkage map. Here, people collect the different section of soybean, and then they tap the natural variation. They collect accession from all over world, and they phenotype them. And those cultivars which are having different phenotypes, response to some disease, tolerance to drought, the yield, and many parameters. If there is variability, then they sequence using genotyping by sequencing like this, and then they construct a genetic linkage map. Okay, so and pin a specific phenotype to a genomic location. This is one of the example. This paper is published in March 29. They have constructed the first SNP-based linkage map using genotyping by sequence to map a male sterility gene on in leaf chicory. Okay, so this is a leafy vegetable wherein they wanted to map which is the coding region of the genome which is responsible for male sterility, and they have created a population by using this way. They have two parent, contrasting parent, and then back crossing they have generated these lines, and then in these lines they have done genotyping by sequencing, and they phenotype each line. Wild type obviously have proper pollen, and mutant have distorted pollen, which is giving rise to a male sterile plant. And after genotyping by sequencing, they found out that there is a specific region on linkage group 9, which is corresponding to a protein coding region, which is coding for a transcription factor called as MYB transcription factor. And there is a mutation in this mutant line that is leading to a male sterility. So using this kind of approach, you can pinpoint which region of the genome is corresponding to a particular phenotype. There are thousands of such papers available. Just for an example, I have taken this one. Apart from genotyping by sequencing, you have transcriptome sequencing application. Wide array of applications can be covered by using transcriptome sequencing. And in my lab, we have done transcriptome of more than 5 different medicinal plants, de novo transcriptome assemblies, and we are working on isolating different candidate genes which are involved in biosynthesis of critical secondary metabolites. Okay, so overview of transcriptome data analysis involves you first acquire data, do the filtration, then do the either de novo transcriptome assembly or reference-based transcriptome assembly if the genome is available, and then do the counting of the reads which are mapped on exons, and then do the differential gene expression analysis. This is a pipeline all the tools which are available for each of these steps, they have been mentioned in this diagram. Okay. So one example, I'll just explain how the differential transcriptome analysis is helping in identification of secondary metabolite synthesizing pathway genes. Okay, so this is an example from opioids, which produces the camptothecin, which is an anti-cancerous drug. What they did was they generated an RNA sequencing data of hairy root and cell suspension culture, and then they analyzed, and they could ultimately identify different transcripts which are involved in this pathway. So this was possible because there was next generation sequencing available, and this has been published in 2013. Okay, so the pipeline is here, and it is not important to go through all the steps, but ultimately I just wanted to emphasize that even if you have a plant whose genome information is not at all available, still you can generate a huge amount of genomic resource in terms of transcripts and identify specific genes which are involved in pathway. Okay. Another application of NGS is identifying alternatively spliced transcript. So alternative splicing is a mechanism created by cell to generate a huge amount of protein diversity. Okay, same gene can be alternatively spliced to give rise to two different type of proteins. Okay, so this is an example in Brachypodium monocot, which is also considered as model plant, and they identified a genome-wide alternate splicing landscape modulated by plant-virus interaction. So they use a PME virus to inoculate or infect to this Brachypodium plant, and then they sequence the transcriptome, and using a routine pipeline they identified alternatively spliced transcript. So ultimately they found out that more than hundreds of immuno-related genes which were alternatively spliced. So you will surprised to see that more than hundreds of genes were alternatively spliced because of the virus infection. Okay. And most of them are NBS, LRR, resistance genes, then RNA silencing, splicing associated factor. And they cloned one of the gene called as SCL33, which is serine adenine rich splicing factor, identified multiple novel introns. Okay, so they found out that there is intron retention in this SCL33, and there is increase in the length of these transcription factor by 219 amino acids, and that will be involved in the virus plant interactions. Okay, so this was possible because of next generation sequencing. You can pinpoint which are the specific genes which are being alternatively spliced in a particular treatment. Okay, so another example or application of small RNA sequencing, NGS is snRNA sequencing, small RNA sequencing, and in plants, people have reported huge amount of literature on different microRNAs and their role in specific uh, phenotype or specific developmental stages. Say, male 172 is involved in chloride development. This one is involved in male sterility, and so on. So uh, there have been a number of reports which says that these are the candidate microorganisms which are involved in drought tolerance. Okay, so the antiviral resistance and so on. So isolating these microRNA and cloning and sequencing is very difficult because the size of these small RNA is 21 nucleotide, very small. So isolating and cloning is very difficult, but you can sequence them by using next generation sequencing. So to sequence them, what you can do is first isolate total RNA, then do the site selection by using, say, acrylamide gel electrophoresis, and isolate a portion of the acrylamide gel, which is around 17 to 25 nucleotide, and then which will be enriched in a small RNA, and then ligate the adapter, and these are the chain adapter, and acquire the sequence like this. Okay, and then after the sequencing, you can filter the adapter sequences and try to map the small RNA sequence onto the genome, and then find out which are the microRNA which are those are overexpressed or underexpressed in a particular treatment, and then try to find out their protein coding targets by using various tools. And if it is matching, like if the microRNA expression is elevated and their corresponding target gene expression is lower, then you can have a pair, and that is involved in a particular phenotype, so that can be reported. Okay, so apart from this, there are many other applications such as genome-wide transcription factor binding site analysis, which is also known as DNA as one sequencing, phylogenetics and ecological studies, single cell genomics, metagenomics that already I mentioned. And people have deviated from normal sequencing pipelines, and they have come up with variants of next generation sequencing. And for specific application, they have been using these type of different next generation sequencing techniques. So this was about the overall idea of next generation sequencing. Now I will try to summarize what we do in our lab and how we are using this next generation sequencing and data science to understand different aspects of myself. Any doubts so far? Anything you want to add? Sriram, is it fine?
Sir, I think you can continue, and participants will ask their queries at the end.
Okay, great. Okay.
Yes, sir. Thank you, sir.
I have used this transcriptome sequencing for many projects, but I just only two projects and briefly summarize how we have used these sequencing platforms to answer the question in our mind. So first one is elucidating bispecific pathway, and the second one is effect of on the osmotic tracing fingerling. Okay. So Plumbago is an aptoquinone, and it is being reported for very good against a number of cancers. Okay, and this has been produced by a medicinal plant called as Plumbago zelenica and many other Plumbaginacea members. It has a potent anti-cancer activity. More than 1,000 papers have been published using plumbaging to control these all cancer types. And not only there are reports, but the molecular target, how exactly plumbaging is modulating these inhibitions of cancer progression, and how exactly at molecular level it is been regulating the progression of these cancers is being very well understood. However, the biosynthesis of this naptoquinone plumbaging in Plumbago zelenica is till date nobody has looked out how the plants are being synthesizing these plumbaging. Can we synthesize this plumbaging in vitro inside the lab, devoid of plumbago zeylanica? Apart from these plumbaging, there are many other naptoquinones which are pharmaceutically important, and all of them, how they are synthesized by plants, is still unknown. Okay, so our aim was to understand how this plumbage is being synthesized by plants, what are the biosynthetic pathways, which are the genes which are involved in this pathway. So the only work in literature available for synthesis of this kind of naptoquinone is first gene, that is polyketide synthesis. So this plumbagine is polyketide, it's a tetraketation. First gene which is reported is polyketide synthase. So in 2006, these authors, they basically overexpressed this polyketide synthase in E. coli, and then they give this starter and extender molecule, and they found out that there are some intermediate form, but not the naptoquinone ring like this, okay, or plumbagine. So this indicates that only alone polyketide synthase is not sufficient for synthesis of naptoquinones in vitro system. So obviously there are some downstream genes which are not known, and we wanted to understand what are those genes and how we can synthesize plumbagine in yeast or some other system. So in our department, only the same polyketide synthase was being overexpressed in tobacco, and some few genes are there in tobacco which could synthesize this naptoquinone but not plumbagine. That can be concluded from this experiment that some plumbago specific genes are required to convert these naptoquinones like isocinolone to the plumbagine, and mostly you can see the difference is only hydroxylation. And as we all know that, since you are also from chemistry background, these hydroxylation, oxidation, reduction type of reactions in case of biological system, they are being carried out by cytochrome P450. And our aim was to understand what all cytochrome P450s are there in the plumbago. So for this, we started first the collection of these, then we quantitated plumbagine in leaf and root, and obviously as reported, we found out that root is very rich source of plumbagine, and then we decided to sequence the leaf and root, and then we send the 2 microgram of RNA for next generation sequencing, and we use Illumina HiSeq 2500 platform, and we filtered the reads and then assembled these short reads by using Dino assembly. The program used was Trinity. Since this is a Dino assembly, because the genome of plumbagine is not available, and then we did the functional annotation and differential gene expression analysis. So these are the read statistics. So for tissue type, we had around 50 million reads, which is more than sufficient, and we got an assembly, nice assembly having these many transcripts, almost 2 lakh transcripts. Okay, then we did the normal differential gene expression analysis pipeline, and we could found out that there are almost 2,900 genes which are upregulated in roots. Okay, but obviously we wanted to understand out of these how many are of cytochrome P450. So what we did was we used the query sequence from say tomato cytochrome P450 and analyze what all corresponding transcripts are there in a plumbago transcriptome, and we could found out that there are 10 different full length cytochrome P450 that is being expressed in leaf and root, and we assign them standard universal nomenclature. There is a nomenclature committee worldwide, and we submitted our sequence, and they gave by after evaluating, they gave these names to the plumbago cytochrome P450, and these are the expression values. So remember that plumbagine is highly expressed or synthesized in root. So our interest was to identify which are the genes or transcripts those are upregulated in roots, and we could found out that there are only 2 transcripts which are significantly upregulated in root, and we took polyketide synthase, which is being earlier characterized that is involved in plumbagine biosynthesis, as an control or that is also overexpressed in root. So these are two candidate genes, SIG B140 and B141, and those candidate further we functionally characterize. So obviously the transcriptome data what gave to us is these candidate genes. So further we need to validate whether really they are involved in plumbagen biosynthesis or not. The later part is to functionally characterize them. We can silence them, we can overexpress them. If these two genes are involved in plumbagen biosynthesis, after silencing, what we should get is reduced synthesis of plumbagen. And if we overexpress them, there should be enhancement of plumbagen biosynthesis. Then only we can say that these genes are involved in plumbagen biosynthesis. So later we did that. So initially, first we did a phylogenetic analysis to ensure that they are also clustered along with the reported genes, and these are the two genes which are, we call them as a candidate genes, which are also reported to be involved in hydroxylation by other groups like 81 family, 81B family involved in hydroxylation. Okay, so for apart from that, we did real-time analysis. All the 10 genes, their transcriptome analysis gave us some expression profile, and we did a real time to confirm that whatever our transcriptome data is saying is in concurrence with the real time data, and we found that there is quite good correlation between the transcriptome and the real time data. And these are the two genes, even real time also reported that they are being upregulated in root. Okay. So for silencing, we used an artificial microRNA approach to transiently downregulate these cytochrome P450. We used this Arabidopsis 390A vector, and we cloned our gene of interest like B140 and B141, and only short part that will generate ultimately microRNA was generated, and it will target those P450 genes, and those will be silenced. Okay, so this is an way of transient silencing. This is a plumbago indica, and we cloned our short fragment of B40 and B41 in this vector and transform it into agrobacterium tumefaciens, and then using agroinfiltration, this method is known as agroinfiltration, we harvested tissue after infiltration 3 days, 6 days, and 9 days. So initially we individually silenced 140 and 141, but what we found out that whenever we silence 140, the 141 expression is increased, and whenever we silence 141, 140 expression is increased. Like if one is silenced, second expression is increased. Second is silenced, the first expression is increased, and we were surprised that there is kind of an promiscuity. That means there is a kind of a complementing function. But then later we decided that we should downregulate both of them, and then we built a construct wherein the region of the sequence will target both the genes, and we could find out that after doing the co-silencing, we could downregulate both the genes. So obviously, after silencing, if these are the genes which are involved in plumbagen biosynthetic pathway, we should get a reduction in the plumbagen. So that was checked by using LCMS. So in our department, we have LC cutoff, wherein individual quantity of metabolites can be measured. So there are few metabolites, intermediate metabolites, and this is the plumbagen. And you can see here, the individual silencing showed some effect on reducing the levels of plumbagen, but co-silencing or silencing both the genes at a time has drastic effect or reduction in the plumbagen content. And this indicates that these two genes are involved in plumbagen biosynthesis, at least at a silencing stage. Obviously, the next question was to overexpress. After silencing, there is a reduction in plumbagen content. If you overexpress, you should get increase in the plumbagen, and that was tried again in a transient experiment. So here we found out that there is an increase after 9 days, even at 3 days, 6 days, there was increase. So indicating that these two genes are cytochrome P450s are involved in plumbaging biosynthetic pathway. Apart from that, we also did the docking studies to understand the molecular site of attachment of these the substrates and the plumbaging, and we found out that there is proper binding of these substrates and product at the cytochrome P450. Okay, so all this we have published in a journal called as Phytotherapia. This is a proposed pathway wherein these polyketide synthase, some few more genes and cytochrome P450s have been reported. Okay. So this is one story where we have used next generation sequencing to pinpoint a specific pathway and understand the candidate genes which are involved in biosynthesis of this naptoquinone type of metabolite. So now we are also trying to co-express all these genes in one vector called as yeast vector, P escape vector, and 8 such genes can be co-expressed by using 4 different forms of this vector. So we have isolated all the genes and put it in these vectors, and we are now trying to overexpress everything in yeast. And if everything is true, whatever we were saying, then yeast should produce the naptoquinone, that is plumbaging, and we will be analyzing it by LC-Q top MS. So we have promising results so far, but we are trying to optimize this yeast expression now. Okay. The second example which I'll be discussing is the role of silicon on retaliation of osmotic stress in finger millet. So we supplemented silicon and given osmotic stress to the finger millet, Illusa and Corecona, and we found out that the silicon retaliates or ameliorates effect of the osmotic stress. Osmotic stress was given by using polyethylene glycol, and you can see here the root length, shoot length can be increased in presence of silicon. So even the silicon uptake was also increased after osmotic stress. So we did the whole transcriptome analysis of 4 different treatments, and again we built the DINO assembly and using same Trinity, and did the DINO differential gene expression analysis. And this time we coupled transcriptome with metabolome, and we could found very good correlation. So this is the transcriptome data, and we built this pathway wherein it is very clear that based upon the transcript abundance, we can say that the glycolysis has the genes which are involved in glycolysis were upregulated, and that will be giving acetyl-CoA pool, and even the TCA cycle was reduced. That means the acetyl-CoA pool was not diverted to something other than the TCA cycle, and it was diverted to the lipid biosynthesis. So ultimately, we concluded that the after silicon amendment at osmotic stage, the lipids are being accumulated, or the genes which are synthesizing lipids, they have been accumulated, and ultimately the jasmonic acid concentration was upregulated. So we did the LCQ of MS analysis, metabolomic analysis, and we could found out that there is a proper clustering indicating that the metabolome of each of these treatments is different. Okay, and this is the slide showing the relative quantitation of these metabolites. So we quantitated this metabolite by using mass spectrometer, and there was significant alteration or difference, and whatever transcriptome data was saying, same was complemented by the metabolome as well. Okay, so we built this metabolic profile where the genes which are involved in upregulation of fatty acids and the corresponding metabolites were increased in under osmotic stress whenever silicon was added, and the whole of the pool was transferred to synthesize methyl jasmonate. And obviously we all know that the role of methyl jasmonate in combating abiotic stress. Okay, so this was one of the example wherein we use these omics approaches to pinpoint a specific hypothesis or confirm our understanding about what is the molecular mechanism of silicon under osmotic stress. Okay, so I would like to now summarize whatever we have discussed so far. So these are general suggestions. Not necessarily every one of you have to agree with me, but from my experience, I have listed all these points. So research must be driven by hypothesis than the use of technology. So all you have seen a lot of technological advancement in this lecture, but all those technologies are there and they will change over the years. But whatever research we are doing should be driven by the hypothesis, like whatever we want to understand, and not by the platform or technology. So design NGS experiment with clear objectives and expectations. So not randomly give the samples for sequencing. Beforehand, you need to know what you want to see after you get raw data. Okay, acquired data from reputed and trusted vendors. This is important because in India there are a lot of vendors coming up, and they will do all sorts of manipulation. So acquire data from reputed vendors. So researcher must understand the data analysis pipeline, deviations for his or her own experimental system, and from standard pipeline. So whenever those companies give you the analyzed data, you need to understand what they have done and then build a story on that. So learn at least primary NGS data analysis. I'm sure that many of you are working in many other data analysis tools, but you should also understand these tools that will be a kind of a rewarding skill set for employability. There are many startups wherein if your students have skills of analyzing the data or handling the large data, their chances of getting employed are very higher. So please feel free to ask the question. So there are many blogs and platforms on internet where wherever you stuck in the analysis, you can write there, and there are worldwide communities which will give you answers. I have my own experience. So when all the analysis I have learned on my own, and whenever I am stuck, you can ask to the original author of the paper or these blogs. They will give you answer with the code as well to analyze your own data. So people are really helpful. Only don't hesitate, ask the question. So with this, I would like to acknowledge the funding from BRNS and SPPU, and this is my research team. I have 4 research students working on different aspects, and thank you for patient listening.
Thank you, sir. Thank you so much. Now the session is open for question and answers. I request all the participants to ask the questions. Yeah. Yes, please, if somebody has the question, please feel free to ask the questions.
For chemistry faculty, I can understand because it was completely on biology angle, but obviously few things are also from chemistry. And as I mentioned at the start of the lecture, unless you work in collaboration with some biologists, wonders cannot happen. Yes, there is some question.
Yeah, one question. How about microbe sequencing advances? Yes, sir.
Yeah, the platforms are same, like sequencing platforms are same. Only the thing is the genomic DNA, whatever you will isolate will be from the microbial origin, and the analysis pipeline will be different. Yeah. Yeah, so do we have indigenous
markers in India, available in India?
Markers, I'm not getting what you are saying. So markers means primers. Oh, so
Primers,
yeah. So for whole microbe sequencing, you don't need primers. What the starting material will be your original genomic DNA. They fragment it and ligate the adapters, sequencing adapter, and based upon those sequencing adapter itself, the sequencing will happen. So even if you don't know any sequence information of microbial genome of your interest, that is also fine. So you can do the de novo assembly, and there are very nice tools available. So in fact, sequencing of microbial genome is very, very cheap. And there are many suppliers in India also who can take your genomic DNA or even a bacterial culture and give you the whole draft genome sequence of your microbe. I hope, yeah, my answer is clear.
Yes, sir. Yes, yes. If anyone wants to ask any question, please feel free to ask. Yeah. Okay, I think questions are over. So I request Harshada to propose the vote of thanks. Yes.
There is one more sequence.
So one, yeah. Sorry.
How many indigenous plants have been sequenced? Uh, yeah, so most of these genome sequencing projects require large grants. There are few plants they have been sequenced from India. You might have heard of ICRISAT Institute at Hyderabad. They have sequenced pigeon pea, chickpea, from medicinal plant, from CMAP, they have sequenced Osimum, Tulsi, and few more are there. But the quality of sequencing is little bit lower. ICRISAT, chickpea genome and pigeon pea genome, they are great, but you need a depth of the sequencing to cover at least to the chromosome level. But if you visit any of these databases, there are many a number of plants. Mostly people are interested in sequencing crop plant, obvious for the reason that they are their improvements will ultimately lead to enhancement in the yield. But there are many medicinal plants have been sequenced worldwide, and few of them are from India as well.
Sir, I have one question to ask actually.
Yes, madam.
Because now the funding is the point of concern. So I just want to ask you for your Plumbagen story, means approximate what cost?
Yeah.
Yeah.
So again, it depends upon the type of experiment. Like 50 million reads of transcriptomes are per sample can be without analysis. You can get in around 25,000 rupees. Okay, so if you have two samples, say leaf and root, so around 50,000 rupees or 60,000 rupees is more than sufficient. But you need to analyze your own data. And I encourage, in fact, in my lab, I analyze my own data. So then only you will get some concluding results. Otherwise, these companies, they have some standard pipeline and they analyze with that data. And if you ask some questions to them, they will ask you to pay more to do a customized analysis. So that is there.
Okay, sir. Okay, thank you, sir. Yeah. If we have finished with the questions, I will request Harshada to propose the vote of thanks. Harshada, please unmute yourself. Yeah.
Am I audible?
Yes.
Good afternoon to one and all gathered here. On this wonderful afternoon, I would like to present a vote of thanks. This is the golden opportunity to thank the respected Dr. Mr. Barwarkar sir. Sir, you nicely explained and gave us a brief idea about the topic omics and its application. Sir, in this topic, in this seminar, sir also told about the array of the omics method, the tools for the omics, and give us a brief idea about the history of DNA sequencing, and also told about the very platform like Illumina, Thermo Fisher Scientific Platform, Pacific Biosciences Platform, and the latest Oxford Nanopore Technologies. Sir also told about how much the sequencing data to collect, how much should our reads be, and where we go with the technical or biological replication. And sir also told about the single end and paired end sequencing. And sir, sir also explained the NGS applications in plant sciences And at last, sir told about his paper work. It was a really nice work which you have done. Sir, I personally impressed with your way of presentation and the matter which you have included in your slide. This is so much informative to all of us, and it is. I think it is a complete package of knowledge for all of us for our future study. And sir, we were really enjoying our lecture. Thank you so much. And I also thanks to the whole team of Professor Ramakrishna More College for gave us a chance to interact with such a great personality. And I also thanks to Dr. Varsha Sriram ma'am who gave me a chance to propose the vote of thanks. Thank you so much.
Thank you, sir. Thank you so much. Yes. Now I will hand over this to Rasale sir. Yeah, Rasale sir. Uh, hello.
Hello, madam.
Yes, sir.
Announce the lunch break.
ओके ओके ना सर किती वेळ काय
अर्धा तास.
अर्धा तास पाऊण झालेला आहे
हो.
मग 1:45 पर्यंत
दीड करा.
1:30 पर्यंत ओके ओके नाउ विल लीव अँड देर विल बी अ लंच ब्रेक and we'll meet again at 1:30. Thank you so much. We'll meet at 1:30. Thank you sir. Thank you once again.
हॅलो शिंदे सर.
हा बोला.
हा येतोय आवाज येतोय.
येतोय का आवाज?
हा स्टार्ट करायचं. शिंदे सर.
हा बोला.
हा ओके. Good afternoon to everyone. I'm going to start third session and give brief introduction of third session speaker, Dr. Shinde DR, working as a professor in chemistry at Ramakrishna More College, Akkudi. He has completed MSc in Savitribai Phule Pune University, also completed PhD from Savitribai Phule University, and then he has recognized MPhil guide, PhD guide. He is under guidance awarded to MPhil students, and 20. He has published a high impact factor journal, 20 journal publications. He has presented conference papers in national and international conferences. He also designed many instruments like DC conductivity measurement, gas sensor, photoreactor. He also constructed thermogravimetric analysis. It is also preparing pellet maker instrument by using hydraulic pressure. He also designed colorimeter, simple colorimeter designed by the Dr. Shinde sir. Now he has given many lectures as a resource person in conferences, national conferences, and also he has also coordinator at incubation center Ramakrishna More College, Akkudi. I'm now request to Dr. Shinde sir given a lecture on analytical method development and validation technique. Okay, I'm now hand over to Shinde sir.
Okay, thank you, Gadve sir, for brief introduction. Okay, my voice is clear. Yes, can anybody?
Yes, sir.
Yes.
Yes. Okay. So you are already familiar with me. So
Yes,
next 1 hour, we will explain the analytical method development and validation for quantitative analysis in the research. Okay. So we will start with why analytical method development and validation is required. When we deal with new type of compound and it is to be analyzed, suppose here botany peoples are there, chemistry peoples are there, zoology peoples are there. We want to some evaluate biological activity such as antimicrobial activity of some plant material extract. And whatever the molecule is there, it is first time isolated. So you have to predict MIC, minimum inhibitory concentration, MIC, okay, or the other types of concentration. As the compound is analyzed first time, there will not be analytical method available to you. So you have to quantify it. So ultimately you will require the analytical method. Okay, same is the true with the chemistry peoples. Suppose new compound is synthesized and you are dealing with the synthesis or such a type of activities. And again, for a new type of synthesis compound, you require the analytical method, and without validation, you cannot use any analytical method for competition purpose. We need alternative analytical method for a substance which is more accurate and sensitive. Method is known. Suppose there is a compound X. Analytical method is known, but concentration is low now. And method, whatever the known method is there, analytical method is there, it is not working in this range. So we require more sensitive method, that is which will work at low concentration, and we need to develop, and ultimately when any method is developed, it is to be validated. It is to be validated. Okay, the compound is known to you. Suppose we have to analyze, say, beta carotene sample. Okay, plant material is there, the blood sample or the plasma may be there, and variety of plant materials are there. So as plant material changes, the carotenoids, carotene and carotenoids, this is the difference. Carotenoids represent the mixture of many carotenoids. So as plant material changes, the other interfering impurities changes. Okay, whatever the impurities they present, they are changes, and under this condition also you require changes, and when any change is done in the non-analytical method, again it is to be validated. Okay, so there are many reasons why we require the development of method and the validation of method. So what is analytical method? Analytical method is nothing but it is the set of instructions or the process which is used to find out quantity of substance in interest in the given sample. Suppose we have sample consist of A, B, C, and D. These are the 4 components. And we are interested only in C. Okay, what is the quantity of C? What is the quantity of C? We have nothing to do what is the quantity of A, B, C, D. C is the substance of interest. Okay, so to analyze substance C from the sample or the matrix, we require the chemical process or the physical process. So analytical method is nothing but it consists of the instruction, set of instruction, or the total process to carry out the analysis. Okay, so if you see the analytical method, it may be combination of physical and chemical process. It may be only chemical process, or it may be only physical process. So analytical methods are based on physical and chemical principle, and if both principles are used together, it is the combination of chemical and physical process. Only chemical principles are used, chemical methodology is used, it is the chemical process, and only physical process is used, then it is the physical method of analysis. Okay. So, for example, we have analysis of glucose by calorimetry. Analysis of glucose by calorimetry. Okay. So for example, glucose oxidase peroxidase method is used. So glucose react with oxidates. Okay, and glucose is oxidized. In the process, H2O2 is released, and this H2O2 react with uh, paratoluidine, okay, or other type of coloring substance. Number of kits are available in the market. So phenolic compounds are used to oxidize this phenolic compound, and we get color. So glucose is converted to the some color material. Directly glucose is not converted, but whatever the quantity of H2O2 released, that oxidize the organic substance and we get the color substance. So this is the chemical process and colorimetry. In the colorimetry, we determine absorbance. So it is the physical process. So physical chemical process. Only chemical process. For example, analysis of fatty acid in the titration by reaction with NaOH. By reaction with NaOH. Okay, so this is the only chemical process, acid-base titration, and only physical process. So we have the many methods they based on the physical processes such as the HPLC, GC, spectrophotometry. All they use the physical methods such as adsorption and desorption, or the interaction of electromagnetic radiation with the molecule. As such, there is no chemical reaction, only physical interactions, and these methods are called as a physical methods of analysis. So analytical method is a set of instruction how to carry out the analysis according to physical and chemical method, or chemical method, or only physical method. Okay. Now, particularly as far as the lecture is concerned, I'm dealing with organic compound. We have the organic and inorganic compounds. So the strategies are totally different. Strategies are totally different. So I have selected the example of the organic compound. Okay, we have organic compound which is to be analyzed from the sample. So there are a variety of methods such as chromatographic method. We may have gas chromatography. We have HPLC, high performance liquid chromatography, HPTLC. We may have the spectrophotometric method such as the spectrophotometry and photofluorimetry. Then we have the biochemical method. Okay, biochemical method such as the ELISA, electrophoresis, radioimmunoassay. In that, I can add the electrochemical methods also. They are less common. Okay, radioanalytical methods are also there. Okay, such as the radioimmunoassay or the other type. So we have the variety of methods. Now, the organic compound is given to you, and you have to decide which method is to be used for the analysis of given compound. Okay, so how analytical method is selected? So first of all, we have to see what are the properties of substance to be analyzed. For example, organic compound, when we are saying what is the functional group? What is the functional group? Okay, do functional group is reactive so that we can use the chemical method. If there is no reactive functional group, you have to see, do it have the UV visible absorbance? Okay, it have UV visible absorbance, then UV visible spectroscopy can be used. What is the structure? Okay, so what is the complexity of sample? What is it means? It is single component, that is pure, or it is impure. And if it is impure, how many other substances may be present in that 2, 3, 4, 5, 6, 7, 8, like this. So the second thing we have to think about, what is the complexity of sample? Okay, from given sample, how many substances are to be analyzed? I have taken the example, suppose A plus B plus C plus D. Okay, these are the 4 substances present, and from these 4 substances, we have to analyze only C. We have to analyze only C. So we are interested in C only. But some other person, he may be interested in B and C. Some other person may be interested in A plus C plus D, and some person may be interested in A plus B plus C plus D. So what is to be analyzed from the given substance or given sample, not substance, okay, given sample is important. Okay, then what are the facilities available? Okay, so we have to analyze C, and you have suppose I can analyze C by GCMS or LCMS, but this facility is not available with you. This facility is not available with you. So whatever the method selected, it is not having any use since you don't have the availability of the facility. Cost of analysis, other factors, time requirement, etc. In research, this is very, very important. Now, suppose you have to analyze, say, 30 samples, and you have to use LCMS. The cost of analysis for one LCMS sample for academician is Rs 1000 at SPPU facility. If you go to the other facilities, commercial facilities, you may have to pay 2,000 to 3,000 rupees per sample. So you have to consider how many samples are to be analyzed. If you say cost 1,000 and 30 sample, you have to pay only 30,000 for the one sample analysis or the sample analysis where you have the availability of the 30 samples. Okay. If you have to pay, suppose 3,000 rupees, the cost is 90,000 rupees. So how many samples are to be analyzed and what is the cost? If same thing is to be done by using the solvent extraction, invisible spectroscopy, you can analyze one sample within area 60. Within area 60. Okay, this is the charge. Or if a visual spectrophotometry is available with you, the cost will be very, very low. Okay, so the cost of analysis is very important depending on your number of samples. Suppose you have to analyze 1 or 2 samples. The cost is okay, 1,000 per sample, only you have to spend 2,000 rupees. Time requirement. Suppose your sample is unstable. Isolated sample is unstable and it must be analyzed within 3 hours. And facility is not available to you. You have to extract sample, you have to go to the, say, SPPU facility. Okay. And if there is a availability of the analyst and the time slot is available, it will be analyzed. Otherwise, you will lose all of your work. So time requirement is very, very important. Some samples are stable for 1 month. Such a sample can be submitted to such a central facilities and can be analyzed. But if sample is unstable, the problem will be there. So these are the 5 important parameters when you have to select the analytical method. You can select spectrophotometry, you can select the chromatographic method or the biological method, but you have to consider all these things. After consideration of this, you can go to the choice of sample. Okay, and I will take a very simple example of the beta carotene. Okay, simple example of the beta carotene is to be analyzed. Okay, so beta carotene is the antioxidant material, provitamin A, and in many food supplement it is added. Okay, so food sample is to be analyzed for the beta carotene content, and you have given the project to your UG or the PG student. What is the method that you can use? So first of all, we should know the properties of beta carotene. This is the structure of beta carotene. Okay, so if you see, we know the structure. Second, it is a non-polar one. It is non-polar. So immediately, if you have a structure and the name, you can use the chemoinformatics, put up the word in the Google search and find out all the information regarding the beta carotene. Okay, so you will use the tool, and by using this tool, chemoinformatics, you will get all the information. It do not have reactive functional group. No reactive functional group. Fourth, it is red in color. Fifth, it absorbs in visible region. Okay, this is the important information that we have obtained regarding the beta carotene. Once information is with you, molecular properties are known, you can go to the sample, you can go to the selection. Okay, so if you see name of the component to be analyzed is known to us. Sample is complex in nature. Why I have put up this word? Sample is complex in nature. Since we have to extract it from food or plant material, and when we extract carotene along with many other organic substances, they also get extracted. Okay, and that's why there is no presence of only beta carotene, but other substances, they are also present into it. So it is red in color and it is having UV visible absorbance, and it is strongly absorbed around 500 nanometer. Structure is known. Okay, so this is the available information of beta carotene with you. Okay, now we have to go to the selection of method, spectrophotometry, GC, HPLC, and the HPTLC. So among the various methods, these are the 4 suitable methods. They can be utilized for the analysis of beta carotene. So first one is the spectrophotometry. Okay, what is the requirement for spectrophotometry? The requirement of spectrophotometry, there should not be other substance. There should not be the presence of other substance in the sample which absorb at the same wavelength of beta carotene. Okay, same wavelength of the beta carotene. Another standard should be available. Okay, so if you think of this, along with carotene, there are a variety of carotenoids. They are present and they absorb at the same wavelength as that of beta carotene. So spectrophotometry cannot be used. So my remark, cannot be used. Now, why I have started with the spectrophotometry? Always start with the simplest method and then go to the complex methods. Okay, always you have to think which is the method that is simply available with me and I can carry out the analysis. Second one is the gas chromatography. What is the requirement for gas chromatography? Substance should be volatile below 250 degrees Celsius and it should not undergo decomposition at the temperature of injection port. Injection port is hold from room temperature to 250 degrees Celsius in gas chromatography. And sample is evaporated in the injection port. So it should be stable. If it undergoes fermentation, you cannot use the gas chromatography. Standard should be available. You should have highly pure beta carotene with you when you want to analyze by gas chromatography. Okay. So do GC can be used? Yes. Yes. GC can be used. Why? Since the sample is complex in nature, in gas chromatography, separation take place first and then quantitative analysis. So GC can be used. Do HPLC can be used? So what is the requirement for HPLC? Proper mobile phase in which beta carotene is soluble. The detector, okay. If you see in the HPLC, variety of detectors are available with HPLC system. UV visible detector is there, then photofluorimetric detector is there, then we have the mass spectrophotometric detector is there, coulometric detector is there. So if you have the HPLC having the UV visible spectrophotometer as a detector, beta carotene can be analyzed. You should have the suitable column and standard should be available. So RPC18, if RPC18 column is available with you, since when you are saying the HPLC, there are the three types of the choices that you have to do. One is suitable mobile phase, another is detector, and third one is the column. Okay, so column. Variety of columns are available. Ion exchange column is available. The RPC8 is available. RPC18 is available. N-capped columns are there. Okay, non-polar columns are there. Polar columns are there. So variety of columns are available in the market. And if you want to analyze beta carotene, it's non-polar in nature. And if you have the availability of RPC8 or RPC18, okay, RPC8 or C18, you can do the analysis. HPTLC, okay, you should have availability of HPTLC equipment, and HPTLC equipment usually have the UV visual detector. So your compound should have UV visual absorbance. So beta carotene has UV visual absorbance, and if HPTLC equipment is available, you can carry out analysis by the HPTLC also. Now we have the choice. How many choices are there? You have three choices. You can use the gas chromatography. Okay, you can use the gas chromatography. You can use HPLC and you can use HPTLC. Now from these three, you have to select one. So among these three, the most simple is HPLC. We have to not adjust many more parameters. So HPLC is the simplest. The next one is the GC, and next one I will give preference to HPTLC, not GC. If HPTLC is available with you, you can do the HPTLC analysis, and with HPTLC, many samples, 8 to 10 samples can be analyzed simultaneously. In GC, you can analyze 10 samples one by one, but in HPTLC, if you have a large number of samples, equipment is available, you can use the HPTLC, but accuracy of HPTLC is less than GC and HPLC. So what is your aim? Okay, what is your aim? You require highly accurate analysis, or error up to 5% is allowable. Depending on that, you can do the analysis. HPTLC is less accurate than HPLC and the GC. And the last preference I will give to the GC. Since GC, you have to adjust the temperature and all these things, and we have to wait many more parameters to be analyzed. The gas flow parameter, gas selection is there, you will require helium or you require the argon. So cost is high as compared to the HPLC. Okay, so I will take the HPLC. Okay, so I have discussed already this part. The spectrophotometry is not the viable option since there are complex, sample is complex in nature. Chromatographic methods are suitable. So GC, HPLC, HPTLC, all they can be used. Biochemical method, they are not suitable for the analysis of the beta carotene. Okay. So I have decided I will go with the HPLC. I will go with HPLC. HPLC system is available with me, so I will go with HPLC. So what should be available with HPLC system? Column RPC18, or I already told you RPC8, reverse phase C8. I will explain this term later. Reverse phase C8, detector UV visible, mobile phase polar, and standard beta carotene standard. It can be prepared in the lab and characterized, or you can purchase the certified standard. Okay, beta carotene you can extract from the carrot or any of the green vegetable which is rich in beta carotene. Perform the column separation. After column separation, record the UV visible spectrum of each of the red colored fraction. One of the fraction will match with standard UV visible spectrum, that will be of beta carotene. So then carry out LC-MS analysis or the MS analysis so that you can compare the molecular weight. Okay, so lab prepared and the well characterized standard beta carotene can be prepared, and I have prepared at my PhD work. Then availability, that is the certified standards are available in the market such as Sigma Aldrich, but the cost is very high. For the 100 milligram, the cost may be around 10,000 rupees. 100 milligram, not gram. 100 milligram, the cost of beta carotene standard is around 10,000 rupees. So many times you can prepare your own standards and you can analyze, and otherwise you can use the certified standard. Okay, now why the C18 column choice? This is the part of analytical method development. Okay, once instrument we have chosen, the first upon the method we have chosen, then why the C18 column? Since if you see beta carotene, it is non-polar in nature. RPC18, reverse phase, it is non-polar in nature. And non-polar compounds, they are very easily separated on non-polar column. Once column is non-polar, we require polar mobile phase. We require polar mobile phase. In chromatography, it may be HPLC, it may be TLC, or it may be HPTLC. One general rule is there. If stationary phase is polar, such as a silica gel, you require non-polar solvent. Okay, stationary phase and mobile phase, they must have opposite polarities. Stationary phase and the mobile phase, they must have opposite polarities. C18 is non-polar in nature, so stationary phase, non-polar. What it means that we require polar mobile phase, and polar mobile phase such as methanol can be used for the separation of beta carotene for the quantitative analysis of the beta carotene. Okay, why UV visible detector? Already I told you that the characteristic of beta carotene, the characteristic of beta carotene is that it has a very high UV visible absorbance, particularly in the visible region around 500 nanometer, and hence we should have the UV visible detector. Okay, so these are the requirements that we have set. Then we have the method development procedure. We have the method development procedure. So first upon, we require the standard. Okay, we require the standard, and some arbitrary concentration we will select. And suppose we have selected some arbitrary concentration, 0.05 milligram per ml. We have prepared the standard of this. Okay, and arbitrary flow rate of the mobile phase 1 ml per minute. When you deal with HPLC, mostly 1 to 1.5 ml flow rate is used. 1 to 1.5 ml. You can start from 0.5 ml to 2.5 ml per minute. With analytical column, you can go with such a flow rate. But ideally it's 1 to 1.5 ml per minute flow rate of the mobile phase. So equilibrate column with mobile phase before experiment. That is, we have to pass the mobile phase through the column. Okay, we have to pass the mobile phase through the column before experiment at least 3 hours. Okay, so that the column is well equilibrated with the solvent. Analyze standard and observe chromatogram for retention time, number of peaks, and shape of peaks. This is very, very important when we deal with the HPLC. Retention time, number of peaks, and the shape of peak. So tomorrow we have the hands-on training on HPLC. At that time, I will explain some of the parameters such as retention time. Okay, time being you have to bear with me, or you are already familiar. Okay, so what we are saying, we have used the standard. We have used the standard. So if standard is there, there should be the single peak. There should be the single peak. What single peak indicate? Your standard is pure. Retention time, it should be around 10 minutes, and in most of the cases should not be more than 20 minutes. Okay, this is the ideal time. Even you can go up to 30 minutes. You can go up to 30 minutes. Sometimes very non-polar substances are there, and you can go up to 30 minutes. But if time is greater than 20 minutes, what you can do? You can change the flow rate. And you can use flow rate such as 1.2 ml per minute. 1.2 ml per minute. And whatever retention time which is greater than 20 minutes, it can be reduced and it may be less than 20 minutes. Now, why this thing is required? When retention time is greater, okay, the peak becomes more and more broadened. Okay, broadening of peak take place as retention time increases. And when broadening of peak take place, the accuracy decreases. This is the importance of the retention time. Okay, so retention time, number of peak, and the next factor, very important one, is the shape of the peak. Okay, This is very, very important when you are handling or you are analyzing the sample first time. Okay, so you may get variety of shapes. Okay, usually we get such a 5 types of the shape in the HPLC, and in the method validation and development, the shape of the peak is very, very important. Okay, if you seen the first peak, what it indicate? Overloading of column. Sample is excess. What it means that you have to decrease the concentration. You have to decrease the concentration. This is the flat part. This is the flat part. Okay. Then second one, it is also indicate there is a overloading of column, but it is close. It is not too much high. Again, the same is the case. Overloading of column is there. So you have to decrease the concentration. Okay. Dilute, go on serial dilution. And by serial dilution, obtain the peaks for the standard. The third type of the peak, okay, this is called as a peak is asymmetric, and you can see this is the asymmetry in the peak, and this is called as a tail in the peak, and in chromatographic method, this is called as a tailing of the peak. A point is that why tailing of the peak? Very similar impurity is present in your sample, which is not well separated from your sample. Okay, and it has a retention time very close to your sample. Okay, the fourth one is called as a frontal in peak. Okay, this is the asymmetry. Okay, or I can do like this. This is the asymmetry in the peak. This is the asymmetry peak, and this is called as a frontal. Again, frontal is due to the same reason. There is a presence of impurity, and the impurity is very chemically closely related, and hence the retention time is very close to your sample. And the last one, you have the very good symmetric peak. So for method validation and development, we require such a highly symmetric peak, which is having the triangular nature, Okay, so you have to go on diluting your standard till you will get such a highly symmetric trigonal peak or triangular peak. Okay, so this is the first thing that you have to do. Okay, now what are the method development and validation parameter? Noise. Okay, so you have to run chromatogram with mobile phase only, and record the chromatogram. You will get the baseline and some small peak is observed here. Okay, there is only mobile phase, but due to the very small absorbance or the scattering of light is there, power fluctuation is there, you are getting such a peaks. And this, suppose this is the peak, okay, and this is called as a noise level. Okay, so whatever the peak height you will get only for mobile phase, which is nothing but the noise level. For HPLC analysis, what should be the noise level? It should not be greater than 5 millivolt. Okay, so usually height of chromatogram is expressed in millivolt. So height of the noise level peak for the mobile phase, pure mobile phase, anywhere it should not be greater than 5 millivolt. Okay, this is called as a noise level. Okay, noise should be very, very low. Sometimes if column is exhausted, okay, column is exhausted. One column you can use for only 300 to 400 sample analysis, injections, instead of sample, injections. If column is exhausted, you get the high noise level, and at such a time, you must change the column. Okay, noise level. Detection limit. Detection limit. Suppose noise level is 4.6 unit. You have run pure mobile phase, chromatogram for pure mobile phase, and you are getting the maximum signal strength of 4.6 unit or 4.6 millivolt. Then what is the detection limit? Concentration of sample which gives rise signal strength twice n. N is 4.6 noise level. Twice n. So 2 into 4.6, 9.2 millivolt. So the concentration of beta carotene which gives rise signal strength of 9.2 millivolt is nothing but the detection limit. And you can see here, I have this signal here of 9.2 millivolt, which is clearly distinct, clearly distinguishable from the noise level. Okay. So we should have a concentration used as peak, which is distinguishable from the noise level. Okay, it is very, very important. So what is the importance of detection limit? Below this concentration, you cannot carry out analysis. If sample concentration is less than this concentration, sample must be concentrated and then can be analyzed by HPLC. Okay. Now suppose I have an example here. The concentration of sample I have taken for the HPLC analysis 0.025 milligram per ml, and I got the signal of 244.55 millivolt. Okay, from this you can calculate the concentration. Okay, so what I have 0.025, that gives signal of 244.55 millivolt. So what will be the concentration for 9.2? So 0.025 into 9.2 divided by 244.55, and this concentration is this much. 0.000941. So it is this concentration is very close to 0.001 milligram per ml, or it is 1 microgram per ml. So if I have beta carotene concentration in sample 1 microgram per ml, I can analyze it, I can detect by HPLC. But if concentration is below this, you cannot use HPLC for the analysis. This is the importance of detection limit, quantitation limit. Okay, so in HPLC, we have a peak height, or the area of peak is directly proportional to the concentration of substance in sample. Okay, so this law is hold true over certain concentration only. Certain concentration only. So we have the detection limit DL 0.001 milligram per ml. So at this concentration, beta carotene can be detected, but here there is no direct relationship. Concentration is not directly related to the peak height or peak area. Okay, so I will take an example and afterward I will explain. So quantitation limit, this equation is hold true. Below quantitation limit and above quantitation limit, this equation is not hold true and we get error in analysis. Error in analysis. Linearity range, again same thing is there. Okay, so we have to use calibration curve method. Calibration curve method. And you have to prepare calibration curve where the concentration is in linearity range. So I will take an example, milligram per ml. Okay, so the concentration should not be high than the detection limit. And using the standard, you have to find out the detection limit and the linearity range. So when any sample is to be analyzed of beta carotene, concentration must not be less than 0.005 and should not be greater than 0.025 milligram. If it is less than this, you have to concentrate the sample. If it is greater than this, you have to dilute the sample and carry out the analysis. Same is the case with the peak area. Same is the case with the peak area. Okay. So again, I will put up the straight line and you can find again, same is the true. Whatever the true for the peak height is true for the peak area. Okay. So by both the ways, we are getting the linearity range 0.005 to 0.003. It is at highest limit. So below this, we have the 0.025. So in this region, you will get quantitative analysis very well. You will not get any error involved. If concentration is above this, there much more error will be there, and you will get less quantity than present in the sample. Okay. Now, the next parameter is the validation. We have developed the method. So we have developed method means what we have did. We have selected the flow rate of mobile phase. We have selected the mobile phase. Okay. We have selected the wavelength for the detection such as a 500 nanometer. Then we have decided detection limit. We have decided the linearity range. And now, do it is working accurately? Whatever the method, it is working accurately or not, you have to decide. So what you have to do? You have to prepare again standard. And I have prepared the three standard, 0.012, 0.018, and 0.23. I have run the chromatogram and I have analyzed the chromatogram. Okay, we have analyzed the chromatogram. And from chromatogram, what is the equation we have? Okay, so we have equation y is equal to mx. Y, say, peak area, m, slope, and X concentration. So when we plot a graph in a Excel, you get this equation. You get this equation. Okay, y is equal to 42694x. So this is the slope. This is the slope. Now we know the y. Peak area for sample. You can calculate x. Then x is equal to what? Y upon slope. That is the peak area or the peak height for sample upon slope. And by this way, we have calculated the values. Observed value for the first sample 0012, it is 122. For 180, it is 192. And for 23, it is 0.0223. Okay. So, do these values are accepted or not? Okay. So, whatever the obtained values are there, they are practically calculated. Okay. These are the practically calculated values. And this is the concentration that we expect. So if you post error is less than 2%, for 2% error, this value should be 0.0124. For 2% error, here 0.194, and for 2% error, it is 0.0235. So usually in HPLC, 2% error is allowed. If you're working in the pharmaceutical company, only 1% error is allowed. So here we have decided that in our analysis, we will allow 2% error. So if you see the first value with 2% error, here I have represented, maximum will be 124. So this value is less than this. So this value can be accepted. So if you see now this value, the error is 6.66%. It is much high. It is much high. Again, if you see in the third one, the error is less than 2%. Okay. So in the validation, what we have to do? We have to inject the sample of known concentration, and by calibration curve method from peak area and peak height, again you have to calculate the concentration. The calculated concentration, that is practically obtained concentration, and expected concentration should match within the range of 2%. If it is matches within the range of 2%, then method is validated. Otherwise, method is not validated. So for two sample, it is matching. For the central one, it is not matching. So here there may be error. There may be the error in analysis. Again, you can repeat one. Again, you can repeat one, and you can see the validation of method. Do we get the values as we expect? Okay. So the validation, the one more parameter for the validation is there, is the reproducibility. Do same result is getting again and again, and it must get then and then, method is good. Suppose we have used the calibration curve method. So this is the calibration curve method, and I have prepared the sample of 0.017 milligram per ml, and I have injected same sample 3 times. I get the peak area and peak height, and from peak area and peak height, I have calculated the concentration, and these are the calculated values: 1, 2, and 3. Now you have to decide, do this result is reproducible or not. What you can do? You can go for the statistical analysis. You can go for the statistical analysis. You can do the standard deviation, okay, standard error, coefficient of variation, okay, and you can decide 4122 412.16 upon 45788, you will get this much value. 0.009 milligram per ml beta carotene in sample. Okay, so the point is that when you have to deal with the new method or the new type of substance, you have to develop method, you have to validate method, and only well-developed and validated method can be used for the quantitative analysis. Whatever the methods, all they are reported in the research paper, all they are developed and validated. Now the last point. Suppose the equipment is not available with you and you have to give rise sample to someone for analysis. Okay, so you have to think of all validation parameters. If HPLC sample is to be analyzed, you have to give rise nature of your sample. You have to provide the information regarding the mobile phase, the wavelength, or the detector to be used. Then and then somebody will analyze for you. Sometimes what happens, people phone me, we have HPLC, sir, you have HPLC, and I have to analyze so and so sample. I first ask to them availability of the standard. If availability of standard is there, analysis can be done. Second thing, do you have the information regarding the mobile page? Since I always to tell you the people, I have only methanol. If apart from methanol, mobile is required, you have to provide the mobile page. Okay, you have to provide the mobile page. You have to provide the sample characteristic. There's nothing in chat box. Crude sample also can be used. Yes, crude sample can be used. Okay, but what happens many times in the crude sample, the content is very, the sample number of compounds are very high. So nowadays there is a one method called as a solid phase micro extraction. Solid phase micro extraction. So if sample is very, very good, you can pass through the column, okay, and you can remove highly polar or highly non-polar compounds. Okay, so this is called as a treatment to the sample, or you can do the solvent extraction. Okay, suppose you are extracted in methanol, you are at methanol, extract sample with water, then remaining sample extract with N-hexane, and then remaining sample dissolve in the methanol. So if very crude sample is there, you may have 3 samples, and suppose your compound water insoluble, you can discard this and you can analyze N-hexane fraction and methanol fraction for HPLC. Okay, so for crude sample, very, very crude sample, this strategy can be used.
Sir, if we don't have a standard sample, in what way we can
Sir, you are not audible, sir.
now, sir?
No problem is there. Yes, Sheikh, sir.
If a standard sample is not available, in
Yeah,
what way we
if
Right.
HPLC is available with you and standard is not with you, it is very, very difficult to analyze. They have to go with the GC. Since in the GC universal detector is available, and due to availability of universal detector, at least you will get the approximate percentage. Okay, from the peak area, you can calculate the approximate percentage of your component. Till with GC problem is there, how you will identify the peak of your compound. So with the chromatographic technique, if you don't have the standard, it is very, very difficult. Even it is impossible to carry out quantitative analysis. Since these are the comparative methods, these are not the whatever we are calling as the absolute methods of analysis. So all these are comparative. It means that we should have the standard of known concentration, then and quantitation can be carried out.
Thank you.
Yes, if there are no questions, I will stop my presentation. Over to the Bhange sir.
Thank you, Shinde sir. I'm now.
हॅलो शिंदे सर डॉक्टर कुठे बंद केले आवाज येतोय का रे
Yes, sir, you are audible.
Yes, audible, sir.
ओके निकम सर जॉईन झाले का निकम सरला फोन कर ना निकम सर जॉईन निकम सर ते त्याला 3 वाजता म्हणलं होतं तर तुम्ही त्याला फोन करा हां 3 वाजता म्हणलं होतं त्याला आता ते जेवण करत असते हा फोन कर एक मिनिट वेट फॉर 2-3 मिनिट्स त्याला जॉईन व्हायला सांग ना तर मी त्यांचं इंट्रोडक्शन करतो Okay, next session is a demonstration session. Now in a technical session, we discuss about today one of the important instrument, that is the XRD, X-ray diffraction pattern. Generally, this instrument, the technique is useful for determination of the structure of different chemical compounds, and it is also useful for to find out the particle size of synthesized compound. Now, these are the different equations and different components are involved in this analysis or interpretation of this XRD pattern. And we got the information related to this compound. Now, this is one of the instrumentation part of this XRD instrument, X-ray diffraction analysis, discussed by Dr. L.K. Nikam. And remaining part, small part, is again the interpretation of whatever spectra getting from that instrument, it is analyzed by the synthesizer. Okay, only 10 to 15 minutes, 10 to 12 minutes required for the analysis of interpretation of the spectra, and 1 hour required for the instrumentation and whatever required for the X-ray analysis. Now, that instrument is available at Baburauji Golab College. Okay, the cost of that instrument near about 40 lakh. And it is a sophisticated instrument, and for the carry out to analysis of that one compound, taking the spectrum of this XRD spectrum, it need near about 300 to 400 rupees. Okay, therefore this facility is also available at Baburauji Golab College Center. Therefore, details information about how to pay the charges and all everything is again discussed by the Nikam sir in this lecture. And also he discussed what of the sample, how much amount of the sample need, what are the procedures and what are the guidelines, so everything is also discussing in his lecture. Now, within, sir, I have some meetings, that's why it requires 3 to 5 minutes for the joining.
सरांना वेळ असेल तर आपण इंटरप्रिटेशन अगोदर घेऊन नंतर
नाही आता येतात सर फोन करतो आले
ओके
okay now Nikam sir is coming join the meeting. Nikam sir is working head and vice principal at Baburauji Golab College. Sir completed his BSc and MSc from Noji Wadia College. Sir completed a PhD from CIMET in 2014 under the guidance of B.B. Kale. He sir joined as a lecturer in chemistry from August 1990. So sir has a near about 31-year experience in a teaching of UG classes and 11 year for the PG courses. Sir also recognized guide of Savitribai Phule University. Under his guidance, 4 students are working as a PhD student. Sir has holding various prestigious position in Savitribai Phule University. He is a BS member of the chemistry and also member of 48/3 committee. Sir also member of the restructuring syllabus committee of Mumbai University and Ferguson College, Autonomous College Ferguson. Sir also life member of Material Research Society and Indian Meteorological Society Pune. Sir also life worker of our institute Pune District Education Association. Sir published near about 26 research article in a reputed journals with a citation 590. H index is 11 and I10 index is also 11. Sir organize various international and national as well as state and university conferences or seminar and workshop at Baburauji College. One of the international conference is the FECM is organized at 2016 at Pune. It is having a grand success. Sir also working in various committees in his college. It is a steering committee member, chairman of the steering committee, and he also was the instrument available at Baburauji College. Welcome, sir. Bange sir. Allow to Nikam sir to share the screen. आवाज येतोय सर?
हा येतोय, आवाज येतोय.
हा ठीक आहे.
माझा आवाज येतोय क्लिअर?
हा येतोय काय प्रॉब्लेम नाही.
हॅलो,
हा येतोय ना सर?
माझा आवाज येतोय?
हा येतोय सर, सुरू करा.
चालतंय.
ओके स्टार्ट ना.
ओव्हर सर. हॅलो,
हा चला सुरू करा सर, करते नाही.
So thank you very much for kind introduction. So namaste everyone. I have been asked to demonstrate hello.
हा बोला ना
Thank you Dr. for kind introduction.
सर इको येतोय तुमचा मोबाईल आणि ते दोन्ही चालू असेल ना लॅपटॉप. इको येतोय इको.
Just a minute.
मोबाईलचा स्पीकर ऑफ करा सर किंवा कुठला तरी इन्स्ट्रुमेंट दोन्ही नका चालू ठेवू.
So I have been asked to demonstrate operations of FTIR and XRD. Since this workshop is dedicated for hands-on instrumental analysis as well as interpretation of the spectra obtained in various characterization, we have established sophisticated instrumental analysis facility center in which we provide such as FDIR, UV-DRS, DC, PNC, AS, and XRD. Today's lecture will focus on operations of FDIR and XRD. I know that it is not possible to get the details of hands-on since this virtual mode will not give actual feeling of handling of that. We have to find some way out under case circumstances. So in that case, I will stop in between and I will try to explain the important steps of operation. In the first operation, that is, our college is having FTI of Simon Jumek. Now you must have experience we have taken FDIR using long time back. We have to prepare again mixture of usual mode and sample, and then you have to take FDIR. Later on, KBR disc or KBR, you have to mix with the KBR and you have to make a pellet of KBR, and then you have to take FDIR. But now advanced technology, it is not necessary to use usual mole method, but for hello, hello,
हाँ सर.
Yes, sir.
hello.
हो सर, आवाज येते है.
Do you listen me? Someone should.
Yes, sir. Yes.
Yes, sir.
Hello, Nico sir.
Of this ATR device. Purpose.
काय अडचण आहे ना?
Hello.
व्हिडिओ शेअर करताय ना तुम्ही? व्हिडिओ शेअर करताना काय अडचण येते का?
Hello.
हॅलो. कनेक्टिव्हिटी वीक आहे का तुमची? जरा हे थोडं आवाजामध्ये फरक पडतोय.
एक मिनिट, मी जरा हॉटस्पॉट जोडतो मी.
आणि व्हिडिओ शेअर करायला का तुम्ही? काही नाही अडचण का व्हिडिओ शेअर करायला?
Okay. Now, do you listen me properly?
Okay.
So ATR stands for attunable total reflectance. We'll not go in details about mechanism of this ATR, but we'll see the convenience and the easy sample preparation using this ATR thing. For few samples, particularly organic samples, you have ATR pellet method, but most of 99.9% samples show you the video recording on hands-on or operations FTR. For that, I will share my screen. Is screen visible?
एक मिनिट. व्हिडिओ दिसते म्हणतात का?
So today, we'll begin with FTR demonstration.
सर व्हिडिओ आणि सुरू नाही झाला.
Yes, sir.
ठीक आहे.
हॅलो.
व्हिडिओ शेअर करताय ना तुम्ही? कुठे आहे तेव्हा? मागे
Sir, it's visible.
विजिबल ना? ठीक आहे. ओके.
Yes.
किती झालं? आणि निकम सर विज्युअल आहे. सगळ्यांनी आय रिक्वेस्ट ऑल पार्टिसिपेंट पिन अप द निकम सर प्रोफाईल. ओके सर. आवाज आ रहा है? आवाज आ रहा है? चाय निकल ना आवाज येत नाही ना इथं आवाज आ रहा है? निकम सर हॅलो निकम सर हा व्हिडिओ दिसतोय पण तुम्हाला त्यावेळेस आवाज तुमचा कमी व्हिडिओ मधला येत नाही तर तुम्हाला एक्सप्लेन करावं लागेल तिथे थोडं काय करा सर व्हिडिओचा आवाज कमी करा आणि तुम्ही स्वतः बोललो तर मग सांगता येईल तुम्हाला कुठली विंडो तुम्ही दाखवताय इन्स्ट्रुमेंटचा पार्ट कुठला दाखवताय व्हिडिओचा आवाज येत नाही ना निकम सर तुम्ही सायमिटली एक्सप्लेन करा कारण व्हिडिओचा आवाज कमी येतोय
ओके
नाही सर येतच
Do you listen me now?
हा येस येस तुम्ही ते एक्सप्लेन करा सायमिटलीसली
Now this is process of background correction. This is again you have to first start the instrument, and for starting the instrument, first process is initialization. And in initialization, there is a background. You take FDR background. This FDR background is used for subtracting peaks due to background. Now, since this FDR is taken in open air, you eliminate or you subtract peaks due to background. So this is background spectra. This background spectra is eliminated from the FDR of sample. So it is essential to take a background before you go for FTR of particular sample. If you don't take background, instrument will not. So let us see loading of sample. It is very simple process. The sample is very simple. You can take FTR of liquid, FTR of film, FTR of glass material, FTR of powder, even FTR of crystalline material. So it is very handy and is very convenient. Taking FTR using this instrument is very simple. Once initialization process is done, only this much quantity is required. Even in few milligram quantity, you can take FTR. So it is ready to take FTR. Now you have to fill the information here. You have to, in comment box, you have to write sample code. For example, it is demo. And now you have to give path for data file. Now this file generated is of SMF extension. It is this file generated automatically. So you can give path. So I have created a folder on desktop for demonstration. So once path is created, next job is very easy. So this path is created. Now you have to just click on sample. FTR will be ready. Now you have to do number of scanning steps. So suitable scanning step, you can go up to 30 or 25 scanning cycles. On the right side, there are parameters and some setting of FTR. This is set by technician. Now here you can see transmittance is more than 100%. For that you have to go for baseline correction by selecting 0, or transmission will be below 100. So this is below 100%. So you have to take care that transmittance should be below 100. What then we go for baseline correction? We can go for smoothing of data. If peaks are not well resolved, you can go for smoothing. Now you have to take care that smoothing should be optimum. Because of smoothing, there should not be disappearance of small peaks. So you should have a control smoothing or optimum smoothing. You should not go for excessive smoothing. Otherwise, small peaks will disappear. So this is where, if it is essential, you can go for additional smoothing. Because in atmospheric correction, there is a minimum smoothing is given already, 5 point. So essential manipulation is atmospheric correction. And then additional manipulation like baseline correction or smoothing is additional manipulation. Now here we can label the peaks using autocursor. It gives wave number of each peak. Now this is print. So we can give print of this FTR. So if it is demanded, now one thing here, you can set the scale. Now see y-axis, some is not visible. So I can set it for 42. So we can provide print of FTR. We can select a suitable template. Now all data is on only one page. So I will take print of only one page. So this is one way of giving data. Directly we can give print. On that print you will get graph as well as data data is required for quantitative analysis or quantitative interpretation. But for functional group interpretation, we need only peak and intensity. On that basis, we can interpret this FTIR by using IR characteristic table. Now, second way of giving data, we can give data in ASCII file as well as SMF file. So we have to go to export and ASCII file will be generated. Now this ASCII file can be opened in Excel using proper steps, and from that Excel we can copy data to any Excel software, so graph software like Origin or Sigma or whichever software is with you, and we can plot the graph. So this is about operations of FTIR, how to take FTIR, how to make sampling, and how to provide data either in print format or either in data format. This SMF file, it can be opened in only software provided by company, that is IR solution. It cannot be opened at other PCs. So this is just only for if you want to again generate the ASCII file, you can take that data to our center, and from that SMF file we can generate again ASCII file. So for that you have to keep this SMF file with you, and ASCII file can be used for plotting graph in various softwares. So this is of FTR. If you have any doubt, any problem, you can ask me about FTR. Then we will go for XRD.
Hello, sir.
You listen me?
Yes, sir.
Was it audible and visible?
Yes, sir. Am I audible to you, sir?
Yes.
Sir, can we use the liquid sample in this?
Yes, yes. You can take FTR of liquid also. Just you have to put a drop on that diamond surface, and directly you can go for FTR. So using AR technology is very simple. There is no any critical process sample preparation like in Newjol or KBR. You know that in KBR case, if this KBR plate becomes fatigue, we cannot take FTR. This should be clearly transparent in case of KBR. And nowadays nobody use Newjol.
Okay, thank you, sir.
Anyone else?
Sir, there is one question in chat box.
Chat box, okay. I will go to chat box. If mixture, yes, because the instrument does not recognize whether it is simple or with the compound. Job of instrument is only take the FTR. You will get well-resolved peak. From that well-resolved peak, you can interpret which type of mixture it is or which type of function groups of mixtures are present. And as you all know, a single characterization will not interpret complete presence of mixture or presence of single compound. So you have to combine interpretation of various characterization, and then you can go for confirmation whether it is a single compound or whether it is a mixture of compound. But FTR will provide so which type of function groups are present, which type of bonding is present. This can be interpreted using FTR spectra. So there is no any problem in taking FTR of mixture. Anyone else? How much sample is needed? Very little quantity. As you have seen, that surface of diamond crystal should be covered. That much sample is needed. Even in few milligram, we can take the FTR. This is one of the advantage of this technology. Very little quantity also can be sufficient to take FTR. Anyone else? Shall we go for next demonstration? If there are no questions on FTR. I shall go for next demonstration. Is it? So shall I wait for some still more questions to come?
Yes, sir. Next demonstration.
Yes, sir. Go to next
We'll go for next
demonstration. Become, sir.
Thank you.
Go to next demonstration.
Now, next demonstration in on X-ray diffraction.
Yes, X-ray diffraction.
At our center, we have got instrument of Rigaku. Model is Miniflex 600. Now this is a tabletop model. In XRD, sample preparation is again very simple. For sample preparation, you have to use either a glass plate or aluminum plate with the window is provided for films or for glass material. So I will begin with the sample, and second part is taking XRD. So let us go for first part, sample preparation. So you can see this is a glass plate and area is defined. In that area, you have to spread the sample uniformly. If you got sufficient quantity of sample, you can cover the total area provided for sample.
हा ते शेवटी घेऊन या,
If
at the end ला
not,
घेऊन या.
even little quantity also can be used.
At the end discussion मध्ये घेतो मी
It
ते.
will affect on only the intensity of the peak.
नाही नाही हा, बघितलं.
If quantity is less, if quantity is sufficient, this is second
त्याच्यामध्ये घेतो आणि
sample holder.
बरोबर बरोबर ते मी chat box मी
This is of glass.
chat box मध्ये ते शेवटी घेतो.
Second sample holder.
हा मी लक्षात ते आता ते
Just a minute.
माईक.
This is second sample loader of aluminum. So we can stick samples on this sellotape, either glass samples or film. I will just demonstrate how to again fit the glass samples. This is again glass sample. So this way you can fit the glass sample on sellotape, and this is ready for inserting in X-ray machine. Second, for powder samples, you can spread sample over that area. As far as possible, you can cover total area so that you will get well intense peak. If sample is not sufficient, even with minimum quantity also we can take XRD. Sample quantity required about, depending upon the volume size, around 500 milligram sample is sufficient if it is voluminous, or 1 gram sample is sufficient. Only quantity should be enough to spread total area. Even if not, quantity is not sufficient, we can take XRD, no doubt. So this is compound is spread uniformly on glass plate. Next part is instrumental operation. Now this is X-ray machine. Now you can see the lamp is on. It indicate that X-ray is on. Now you have to open the shutter. For that opening shutter, you have to unlock it. Once you unlock, X-ray will be off. So for opening the shutter, you have to unlock it. So yellow button you have to press for unlocking. So now it will be unlocked. You will not hear the beep. Once it is unlocked, you will hear beep continuously till you lock it again. So it will blink and it will beep. So X-ray is off now. This is what right side there is a detector, left side is X-ray source, and in between there is a clip provided to insert the sample holder. So in that clip I will insert the sample holder. Now this is what it is ready for X-ray scanning. Right side is a detector. Now it rotates in desired range of 2 theta. Sample also rotate as well as detector also rotates. Source is fixed. Only the rotation of sample holder as well as detector. Now we can take XRD. So here, this is a software for taking XRD. Now there are various steps. These are left side. All conditions are given. Most of the conditions are set by technician. We can change the scanning rate. We can change scanning range. We can change step size. Essential for starting this instrument, first you have to go for already it is been done. But you go for aging, then you can use you can take XRD. Exit control is to manually you can stop the lamp. Now it is template for XRD. So this is what template. Already 7 sample has been again scanned for XRD. Now I will select 8 sample. We'll go to destination. Now you label the
निकम सर
हाँ
कनेक्टिव्हिटी वीक आहे का थोडी नाही ना ठीक येते आवाज
नाही आता मी हॉटस्पॉट लावलेला आहे
चालेल चालेल ओके नो प्रॉब्लेम येते आवाज
Now if you want to stop this after this scanning, if it is last scanning, you can click on stop. Now this is what where you can done setting of parameters. Now 20 to 80, step size is 0.2 and scanning speed is 2 degree per minute. And at the footnote, you will get time required for the scanning, 30 minutes, 20 seconds. Now depending on scanning speed, the time required scanning is changed. So we can have minimum angle 3 degree, maximum angle 90 degree. So we can have a maximum scanning range from 3 to 90 degree. Minimum step size is 0.1 degree. Minimum speed is 1 degree per minute. Now here I have set for 2 degree per minute. Now once it is done, so here speed is 2 degree per minute, step size is not 2 degree, and range is 20 to 80. Now just you have to click on run. So here scanning has started. It will record the XRD. Okay, we'll go for next part. Now once scanning is over, you will see the X-ray spectrum on screen. This is what XRD, this is active one, latest taken. And you can see in gray shade, earlier taken. On right box, you can see collected data of sample 1, 2, 3, 4, 5. The last one is sample 5. You can put manually X-ray source off using exit control so that you can shut down the instrument. Now, so you may identify which sample it is. It is very well known XRD pattern. Will you be able to tell me which sample it is? Anyone from you? Now you can save this. It is saved automatically. Now data is saved in 3 format that I will show you. See, there are 3 formats. First is arrays format, then ASCII format, and third is raw format. So arrays format is good for plotting. So you have to produce data in Excel format, Excel file using arrays format. Now there are proper steps to open arrays file in Excel, and once data is generated, that can be copied in graph software available. Other types of file like raw file, ASCII file, that can be used for directly XRD software like PDXL or Expert or various software which are available for directly XRD analysis. Now this PDXL software is provided by Rigaku company, which is complementary software. So all it is not completely operational. Only few steps are operational. Now here we can plot the graph. You can obtain graph using this software. So you can open any file, either RS file or ASCII file or even raw file. So you will get again PDF of this file. You can label each peak, which gives 2 theta, D value and intensity. We should create report is created in PDF format as well as Word format. This is Word format. Below this peak list, which gives 2 theta, D, height, FWHM, which is required for particle site analysis, intensity in various units like CPS or weightage. You will get total information when analysis is done, sample name, okay. This is about XRD operations. If you have any query or any doubt, kindly feel free to ask.
Participants, if you have any query regarding the XRD, you can ask.
Hello, sir. Am I audible?
Yes, you are audible.
Sir, which kind of the information we can obtain about regarding biological samples by using this technique?
Now, whether the sample is biological or it is inorganic or organic, from XRD we will interpret what is the crystal structure pattern. Now there are 7 crystal systems. Out of that, whether it is a cubic, tetragonal, orthorhombic or monoclinic, like that you can find it, as well as even space group can be found. There are 32 systems in 7 crystal types. So everything can be interpreted from X-ray diffraction pattern. So there are softwares and there are techniques for interpretation. Depending upon which software is available, according to that we can go for at least crystal system or crystal type or space group, and you can go for which types of planes are present. That is called HKL indexing, 100, like that. And from that we can interpret which type of Bravais lattice is present. You know that in case of cubic, there are 3 types of Bravais lattice. So accordingly we can, whether it is body centered, face centered, or primitive, based on that which planes are present, you can interpret whether it is which type of Bravais lattice are present. So this information can be interpreted. Second thing, if you have got what software like JCPDS data, you can compare 2 theta values with the JCPDS data available And if this 2 theta value matches completely with the JCPDS data, from that we can say that your compound is having that structure and that type of crystal systems. You must know the name of the compounds. For example, it is zinc oxide. Earlier spectra which I have demonstrated is of zinc oxide. Now you can see the various JCPDS data is available for zinc oxide, for hexagonal as well as tetragonal system. So from that we can see whether zinc oxide which you have synthesized is either hexagonal system or tetragonal system. You can have 2 theta value with you, and from this 2 theta value you can compare it with the JCPDS data. And while comparing in JCPDS data, first 100% intensity peak should match. Maximum intensity peak like 100%, 50%, 60%. The maximum intensity peak should match. Some intensity peak which are very weak or which are very feeble, that may not match. Some peak may disappear. Some peak may not be appear because of the way you have prepared sample. Because in case of nanoparticle substance, some peaks are disappeared. Widening of some planes is observed, some peaks are disappeared. It means these planes are not existing in these nanoparticles. So in case of nanoparticles, all peaks may not be visible or all peaks may not be observed. So for that, you have to take, for interpretation, you have to confirm that maximum intensity peaks are present. And from that, you can say, confirm that it is the desired compound has been formed. This is about interpretation. So you can go for manual interpretation as well as you can go for the interpretation using XRD software.
Okay, sir. Thank you.
Okay. Thank you, Nigam, sir.
This is powder pattern.
Giving...
Even there is one more type of exercise, single analysis.
Nigam, sir.
For single crystal analysis required for all types of analysis. Only for special types of analysis, single crystal analysis required. From that, you can find the distance between the atoms and the positions of atoms in plane. Everything can be determined in retrieval analysis. And for that, we go for single crystal analysis. But for normal interpretation, which type of crystal system is there, what is phase group, which planes are present, which planes are disappeared, what is the particle size, that can be determined, interpreted from this X-ray diffraction pattern. First is it is used for confirmation that desired compound has been formed. That is first important application of X-ray.
Okay, Nikam sir.
Thank you, sir.
Hello, sir. Nikam sir, all is Mada.
Bola.
There is a question from chat box, that is Nilima Devi. She asking regarding the how much mixture of sample, mixture of 2 compound.
How much?
Mixture of 2 compound. We carry out the IR of that mixture.
Yes, from XRD, you can interpret whether it is a single compound, whether it is a mixture compound, isn't it? For example, if zinc oxide contains some impurities, crystalline impurities, you cannot label or you cannot index all peaks. So peaks which are not indexed or peaks which are not interpreted using JCPDS data, we can surely say that it is because of some adulterant present or some other compound present. In case of composite, like ZNO, TiO2, you will get peaks due to ZNO as well as TiO2. So you can label or you can interpret which peak is due to ZNO, which peak is due to TiO2.
Okay.
So XRD now interpret whether it is a single compound, whether it is a mixture of compound, or whether impurities are present in compound.
Now another question regarding
compared to FTR, XRD is quite sufficient to interpret whether it is a single compound present or mixture of compound is present.
Now for IR, mixture is not
even from XRD, we can find percentage of composition. If it is again 2 constituents are present, for 2 constituents, we can find percentage of each constituent from X-ray diffraction peaks.
No, for IR, the mixture of 2 compound is not carried out for the analysis, no?
We can take mixture of compound.
We can take mixture of compound.
Yeah, we can take mixture of compound.
Because
what is the role of IR? Whatever sample is loaded on this ATR, it will take FTR. Now, if peaks are overlapping, then we cannot identify mixture. Like now is C11 peak. is overlapping in both the seed ball is present in both the mixed compounds. So you will get single peak due to seed ball. So we cannot say this seed ball bond is of which or whether it is present in both the compound or it is of single compound.
And another question from Sunita madam. She asking about the preparation of plant sample. For plant sample, is there any other process of sample preparation?
No, now it's up to you. It is up to the researcher. For what purpose you need XRD? If you want to have XRD of leaf, directly you can load a leaf. If you want to again XRD of some constituents of leaf, you have to first isolate the constituent, whether in solid form or in film, in the form of film. You can isolate this and then you can take XRD of that particular constituent or extract it.
Okay, that
It's
is
up to you.
Sir, thank you so much.
Another question regarding sample is crystalline or amorphous for exact analysis.
Yeah, we can identify whether sample is crystalline or amorphous.
Which
if there is
sample
no peaks are observed, you can say sample is amorphous. Even in XRD software, there is a provision to identify peaks due to amorphous nature. We can interpret whether it is amorphous or crystalline.
How much quantity of sample required for the carried out XRD?
As I told you, that total area defined is we need sample quantity which is sufficient to cover that area of 1.5 by 1.5 centimeter. If not, even with little quantity also we can take the XRD, but only it will hamper on intensity of the peak, nothing else.
How much
But
milligrams?
XRD can be taken.
10 to 50 milligram is sufficient?
Yes, even 50 milligram is sufficient. Even I have taken XRD of even less than 50 milligram quantity.
Okay. That is also confirmed. How do we confirm the result? How do we confirm the result?
Okay, we are going to that.
Okay.
Now for confirmation of result, you have to go for manual interpretation or interpretation using softwares.
Yes, this is also very important.
You will have a manual interpretation for cubic crystals or tetragonal crystals. For other crystals, it is not possible to go for manual interpretation, but for cubic and tetragonal, we can go for manual interpretation. Even for hexagonal, we will have a manual interpretation.
Okay, now all questions are more whatever asking in the chat box. Now any question from participant? Live question, unmute yourself and ask question to Nikam sir. Any doubts or query?
You can send sample for XRD as well as FTIR as well as UV to all center.
Any information regarding these rates and procedure for they also uploaded on our website of with the above Jagolab College Sangvi. Therefore, we also refer the rates and how much sample is needed for that. Everything is mentioned in their website. Okay. Sir, the email ID email ID for contact. Chat box मध्ये लिहा.
My email ID is latesh.nikam@gmail.com. LATESH.
Chat box मध्ये
I will put it in chat box.
Okay, here, right in chat box.
Bani sir.
Shall I take leave?
Okay, thank you, Nikam sir, for spare the time for this demonstration of IR.
Thank you all participants for patience listening.